again, welcome, good morning, good afternoon, good evening from wherever you're joining. We've got participants joining us from all around the world, and we're delighted to have you with us for this third Global Nightjar Network meeting. My name is Logan Parker. I'm one of the steering committee members of the Global Nightjar Network, and I'm going to be hosting this morning, and I'll pass the reins to another steering committee member shortly. And we'll start just by discussing some of the logistics of the meeting and how this will work. If you're returning, welcome back. It'll be quite similar to past years, but we do have some new mechanics here. Just off the get-go, you should know that we are recording these 2 days of the conference, and we will make these recordings available. We'll post them both on YouTube and and up on our Global Nightjar Network groups.io site. We're a volunteer-run steering committee, and we do have some programs that we're developing to develop a small grant program, and those can be supported either through donation. We'll put a link to the donation interface in the chat, and we also have a store where we have shirts and stickers for the relating to the project available, and the funds from those also go towards supporting the small research grant. So we have captioning enabled. So if you would like to use captions and you're not familiar with Zoom, if you click on the More icon down in the bottom right hand corner of the screen, you'll see a option for captions, and you can enable those and follow along that way as well. So again, we ask that you keep your microphones muted throughout the course of the conference, other than during discussion periods. There will be times where we have smaller groups that can break out and have discussions. That's just to keep things running smoothly here on our end, because we'll be playing the recordings, so we want to minimize those interruptions. If you need any assistance, you'll see that there are a number of co-hosts. Those are steering committee members. You can view that in the participant panel, and you can reach out to myself or any of them to assist you. We also have a anonymous feedback form, which we can put a link to in the chat as well, and you can submit any feedback that way, which is certainly fine. And I'm going to read through our code of conduct before we get going here. Pull that up for us. Okay, so this is the code of conduct that the steering committee prepared. So we are committed to maintaining a safe space for people of all backgrounds, especially marginalized groups, including Indigenous peoples, racial minorities, and members of the LGBTQ community. There's zero tolerance for bigotry and hate. Racist, sexist, or culturally insensitive comments will result in being removed from the meeting. If you have any concerns at any time, feel free to directly contact one of us via the private message. We're also providing that anonymous form that I just referenced, where you can voice your concerns. Because these are anonymous, we won't be able to respond to those directly, but you will be able to get that feedback to us at the steering committee, which we will review and take those into consideration. The chat is for conference-related questions only. Please hold your talk-related questions until the end of presentations. With that in mind, feel free to have your questions pre-typed. Sometimes there's a little bit of a lag after a presentation ends, but just hold off on entering them until towards the end of the presentation. Depending on the preference of the speaker, questions may be asked or answered in the chat or spoken verbally on camera. The moderator will provide a written transcript of those spoken questions and answers in the chat. For native English speakers, remember that this is an international conference and English is not the primary spoken language for many of our presenters and attendees. So it is encouraged to speak slowly and clearly and enunciate as best you can. Something I need to keep reminding myself of. And please be patient with us conference organizers as we do our best to keep things running smoothly and make this as inclusive a conference as we can. We're very open to feedback, and we'll provide that survey at the end of the conference, or provide a second survey at the end of the conference for all attendees, where you can suggest improvements for future meetings. And yeah, last, let's just have fun and learn about nightjars. Okay, how am I doing on time? Doing okay, that's great. So we'll be getting started in just a moment. So we'll have, we've got 3 sessions today. Sorry for the whining dog in the background. We have 3 sessions today, and we will have some break periods throughout. We also have a plenary talk that will be following our first session. We also have a poster session and a discussion period that will be the last part of the day. And yeah, we'll give you a bit more feedback or a bit more direction on how exactly that's going to run once we get to that second. But that's going to be that period where we can have a little bit more informal discussion and get on camera and see each other. Okay, so I think that covers most of the official business here. And if there's anything that I've missed, I encourage other steering committee members to chime in on the chat and add some more details. But yeah, we will be running all of the presentations are recorded, but we do have some presenters that will be here to answer questions live. So with that, I
have a quick thing. Thank you, Logan. Rather than creating a poll for topics for breakout rooms, I'm unable to create a poll right now. So I'm just popping into the chat. What topics would you like in breakout rooms later today? If you want, you can type in 1 or 2 or another, and we'll do our best to sort of see what folks are most interested in. We don't want too many breakout rooms today, so we'll probably create 2 or 3. So if folks want, they can just type ideas into the chat. Thank you, everyone. Looking forward to the talks.
Perfect. Thank you, Maria. Yes, that's great. Great to get that covered right at the onset. Yeah, we're looking forward to those discussions later. Okay, so with that, I think we're going to move into our first session of the day on nocturnal behavior. Our first speaker is Christian Perez Granados. He's affiliated with Alicante University in Spain. And the first talk of the day is titled Illuminating the Nightlife of 5 Neotropical Caprimulgids, Vocal Behavior Over a Year and Monitoring Recommendations. And with that, we will begin the presentation in just a moment.
Hello, all. Good morning, good afternoon, good night, wherever you are. Thank you for joining to listen in this talk. This is an honor to be the first starting this session about nocturnal behavior. I would like to thank first the organizer for this meeting. Thank you for all your time. I'm Cristian Pérez Granados, postdoc from Alicante University, and I'm gonna talk today about some studies that we carried out the last years with 5 species of nightjars in the Brazilian Pantanal. And I will be giving this talk entitled Illuminating the Night Life of 5 Neotropical caprimulgids, Vocal Behavior Over a Year and Monitoring Recommendations. So these are studies that I developed together with Carl Schussman and Marinette Marquez, both professor at Federal University of Mato Grosso of Brazil, on which I was doing also postdoc like 3 or 4 years ago. So in all our studies, we were using this technique called passive acoustic monitoring that probably most of you have already heard about it. But in case don't, this is a technique that is based on the deployment of sound recorders in the field that you can leave them for weeks or months, even years. So you only have to replace the batteries from time to time. And you can program these recorders to only record during the period of interest that you want. It can be at sunrise, at sunset, during the night, or whatever. And at the end, you have to listen or analyze automatically these recordings. So this is a method that has been employed for different species, as you can see in the picture, but this is almost perfect to work for nocturnal birds, nightjars, and in tropical rainforest, because in both of these situations during the night and in rainforest, this is very difficult, almost impossible to see the birds, and you can only work by ear, so you can use these recorders. So the methods that we have employed for the 5 species is always the same. We had recordings collected from 6 different locations in the Brazilian Pantanal, separating more or less a minimum of 600, 800 meters between them. So trying to avoid to record the same individual from 2 neighboring stations. So the recorders were left there over a year, from June 2015 until May 2016, and we were recording the first 15 minutes of each hour. So we collected tens of thousands of recordings. So we have to make use of some automated software to analyze the recordings and look for the targeted species that we want to work. So in our case, we use Kaleidoscope Pro. This is a commercially available software that, well, for some species, this is working very well, as we will see later. So the first 2 species that we wanted to work were the common and the great potu. Indeed, they have very nice vocalization. And we selected this species because there were very little information about this family, about when they vocalize, about their ecology. So potus are they have been studied just few times, and most of what we know from them comes from anecdotal records, so we want to do something more sound. We also worked with the common pauraki and the little nightjar. These species, they have been more studied, they are much more common and abundant in the Brazilian Pantanal. Indeed, we recorded like more than 100,000 vocalizations for some of these species, which also allow us to analyze why and when they vocalize more, to do something more ecological. And at the end, we have already published the study with these 4 nightjars, so we wanted to do something different. We want to provide something really new. So we wanted to work with the nightjar for which there was less information available. That in our case was the band-tail nighthawk. So what we did here, we have the list of nightjars that can be seen in the Brazilian Pantanal. So there were 15 species taken out. So if you want to see nightjars, the Brazilian Pantanal is a good place, sure. So we have 3 indexes for each species: the number of scientific publications published, like an index of scientific knowledge that we have about them. And we also have the number of observations uploaded to eBird and the number of recordings uploaded to Xenocanto, like an index of public awareness and information available that can be used for future studies, for example, to assess the change in the distribution, develop some recognition, etc. So what we did was to combine these three different indices in once to identify the species for which there was less scientific and public knowledge. And the winner, or maybe the loser in that case, was the band-tailed nighthawk. Indeed, there was any scientific publication before our study, so there was no idea about when they vocalize, when they breed, when they do anything. So here you can see how worked kaleidoscope for the 5 species. So we can see that the precision ranges a lot. So we have low values between 10 and 30% for the 3 more rare species, the common potu, the great potu, and the nighthawk. But we have very high values for the most common species. So this is probably explained because when you have more vocalization of a species in your dataset, kaleidoscope is more able to classify them well. But when they are very rare and you have only some from time to time vocalization, the software is not working so well grouping them together. But anyway, it didn't have any impact in our results because at the end we check everyone one by one, every vocalization saved by kaleidoscope. So we are 100% sure that we were not considering mislabeled detections. The only problem is that, of course, for example, for the common potu, it took much more time to clean the database than for the little nightjar or the common parakeet. But what is more important here is that we found that for the 5 nightjar species, there was a very high percentage of vocalization detected, between 70 and 85% for the different species. So we are pretty sure that we were detecting almost most of the vocalization. So we were able to see when the species are when they vocalize. So the first thing that we did was to describe the DL pattern to see at which hours they were vocalizing more or less, and we found large differences between species. For example, both potus, they have a larger peak of vocal activity during the whole night, but especially before sunrise, but also a second, the slower peak after sunset, but they were quite different for 2 potus. But then, for example, for the little nightjar, the black dots, it shows its maximum vocal activity just at sunrise, while, for example, the common parakeet showed a more constant high vocal activity during the few hours after sunset. And then, for example, the nighthawk concentrated most of its vocal activity just at sunset. So just if you want to look, for example, for the nighthawk, you should go at sunset. But if you want to work with the little nightjar or the potus, you should go at sunrise. However, the annual patterns of vocal activity were quite similar for the 5 species. The 5 species show with its maximum of vocal activity mainly between August and October. So this is probably because during August is when the first rains occur in the Brazilian Pantanal, when the wet season starts. Indeed, during the monitored year, the first rains took place around 20 of August. So usually following the first rain, there is a peak of insect abundance, also of fruit abundance. So probably this is explaining why nightjars start to vocalize mainly from August onwards. That is because this is when they breed during this period of maximum insect abundance. We also analyzed the impact of moonlight, of the percentage of the moon illuminated in 4 of the 5 species. We didn't consider the nighthawk here, and we saw any relationship between vocal activity and moonlight for the common potu, and even for the great potu, this impact was very low. So we could say that vocal activity of potus was not influenced by moon, while, for example, for the little nightjar and the common parakeet, we found that both species vocalize much more during full moons than during new moons. However, what is more interesting is that we analyze that this impact of the percentage of the moon illuminated in vocal activity of nightjar depends on the hour of the day. So, for example, during midnight, the part that you can see in the middle for both the little nightjar and the common parakeet, we can see that the species only vocalize at midnight when there were full moons, because during new moon nights there was no light, so they were probably just perched in the ground, in the tree or wherever. But then, for example, we found that in both species, there was a much higher vocal activity during the crepuscular period at sunrise and sunset when there was new moon. So when the moon was not illuminated, in these cases, the species vocalized much more during these few hours of the night when there was some light. So probably when they focused, when they did most of the foraging activity. So we can summarize that passive acoustic monitoring has proved to be a very useful tool to monitor these 5 species of nightjars that we found large variation between them in the hours in the real pattern of vocal activity, but also the impact of the moon varied between species. However, their seasonal pattern of vocal activity, so their breeding time, was much more similar for the 5 species, and probably this is explained by the period when they breed, when there is a maximum abundance of insects. So thank you for joining, for listening. I'm happy and open for questions now here. You can see also my email here. You can write me if there is any doubt or discuss later in the chat. Thank you.
Fantastic. Yes, thank you very much for that presentation, Christian. And I'm told that Christian is here to respond to some questions. If we have questions, we have a few minutes here. And I see also in looking at the chat, Christian was sharing some references right in the chat, which is really actually very helpful. So thank you for doing that. Okay, so I've got a question for you, Christian. So the Brazilian Pantanal sounds like an incredible place for nightjar research. Do all species that live there, not included in your study, have distinct vocalizations, and is there much known about them?
Well, about the first question, I'm not totally sure, but I guess that most of the, well, sure that all the species have distinct vocalization. I know that some of them, they are more similar to some, even to some kind of owls, like the pygmy owl. I think that there are some nightjars that they are more similar, but for sure you will be able to identify all of them. But maybe you need to listen the sound, not only seeing the spectrogram like we did. And then about the last question, I would say that not. So there is not much known about them. So in the Brazilian Pantanal, for example, most of the species, there are species that they have never been worked before, but there are much more knowledge for species like the common pauraque, some of these that they are neotropical, but they have a large distribution range. So they even reach the United States. So the big difference is if you are working with nightjars that they are very neotropical in the strict sense, or just for South America or the southern part of South America. But if there are the other nightjars with a larger distribution, they are much better known. But then there are some other cases of endemic species just from French Guiana or some of these countries that we don't know almost anything, especially for potus and these even more rare species.
Yeah, I mean, really
And
highlights.
then about the second question.
Oh, yes. Yep.
And then there is this question about why we found some relationship for some species but not others. So probably in that case for the potus, that is the one that we didn't found. Indeed, we found a relationship for one of the species, but it was very weak. So probably it's just a matter that they are very rare, or at least we were not able to detect too much vocalization. So for the potus, we detected like 2,000, 3,000 vocalization over the whole year. So I think that is more a matter of sample size to find a statistical relationship than any other thing. So I cannot say that the vocal activity of potus is not influenced by moon. I think that maybe it's because in the end we maybe detected the potus just in 2 or 3 stations, and you are only recording probably 1 breeding pair per recorder. So even in this kind of study that seems that you are recording and doing a very great job, at the end you are only monitoring how much 1 or 2 breeding pairs per recorder in the best case. So probably I think that it can be a relationship, but it was not found.
Excellent, thank you. Yes, and you've got another question in the chat here from Carlos. Do species producing churring-like calls sing more often on dark nights than those producing more intermittent calls? Or, in other words, could call type explain differences in the degree of adjustment to lunar activity?
Yeah, it can be. I don't know. So the potus, they don't make any kind of churring. They are more. One of them is like. Yeah, maybe it is, but I cannot say with my studies. But for example, we found that the only species would make a longer vocalization. It was highly influenced, but not the potus that they have more short vocalization. So maybe it can be influenced, but for sure, we need much more studies, maybe some kind of meta-analysis. That there are a lot of studies testing the impact of moon on Nightjar. So maybe someone from the group can work on this kind of meta-analysis.
Yes, yeah, definitely plenty of opportunities to collaborate amongst this group. We're definitely covering a lot of ground. You have another question from Juan. I think we have time for this last question. Have you found some degree of variability among vocalizations of the same species throughout the year?
Well, that's interesting. I didn't look for that really. So even if, because for this software, you need to reintroduce some parameters. So he's looking for sounds that meet the criteria. So even if the species, some of them is vocalizing much more longer, much more shorter, maybe I lost it, but I didn't check for that. So whenever I found a vocalization, I just say, okay, this is my species. Take a note for the database, but I didn't check for that. But it will make sense that especially maybe they vocalize longer in some periods of the year.
Excellent. Well, thank you very much. And these questions have been fantastic. So yeah, please keep these questions up. Feel free to continue having discussions in the chat. If you're up to answering more questions, Christian, that'd be great. And thanks again for presenting and for starting this conference for us.
Okay, Matt, thank you all.
Okay, so we're going to move on to our second presentation of the day. This presentation will be done by Inyunam of AP Levantis Ornithological Research Institute in Nigeria. This next presentation is titled "Acoustic Sound Recorders Are More Efficient Than Point Count in Sampling Nightjar Populations in an Afrotropical Landscape." And we'll start that presentation in just a moment.
Hello, my name is Inyunam, and I'm from the Nocturnal Avian Ecology Lab of the AP Levantis Ornithological Research Institute, where we are trying to understand the factors and processes that underline the spatiotemporal distribution patterns of nocturnal bird species richness, or nocturnal bird species. I will be giving a talk titled "Acoustic Sound Recorders Are More Efficient Than Point Counting Sampling Nightjar Populations in an Afrotropical Landscape." Basically, across Nigeria, we've had several studies of birds, or several bird studies, and these studies have focused on species inventory, distribution, community ecology, foraging, physiology, agroecology, and so you can name them. They've cut across various aspects of ornithology. However, the studies have majorly been on diurnal birds, with just very minimal studies on nocturnal birds. So this could be as a result of poor sampling techniques, security concerns, and access to remote areas, unavailability of skilled personnel, and it has tilted conservation, or made sure that most conservation decisions are being made as a result of studies from diurnal birds rather than nocturnal birds. And thus there is a need for appropriate scientific methods and materials to inform conservation decisions that incorporate both nocturnal birds as well as diurnal birds. Secondly, there is the need to standardize or make standardized and efficient methods available for nocturnal surveys and nocturnal bird monitoring. And we ask the very big question, you may say, what method is more effective to sample or survey nocturnal bird species, and in this case, Nightjar populations. So this study was conducted within Amorum Forest Reserve between the months of November and December 2021, and we sampled we used 40 points in general. We had 20 points within the reserve and 20 points outside the reserve. The Amorum Forest Reserve is a wintering ground as well as an area conserved or managed by the AP Leventis Ornithological Research Institute. It is home to over 350 bird species, which is over 1/3 of the bird species found in the country. It is also home to the regional endemic Just Plateau indigo bird as well as the rock fire finch. So the points were stratified and placed at a 500 meters interval from each other to reduce pseudo replication. Within the reserve, we have 3 habitat types majorly. We have the gallery forest, we have the rocky outcrop, and then we have the savanna grassland. So we used 20 Joule-like digital omnidirectional acoustic recorders, and this it recorded birds heard and seen from 6 p.m. to 6 a.m., which is astronomical dusk to dawn. And then we used the open source software Audacity to aid the identification, which was done by a single observer to eliminate that bias. Every time we recorded a new individual, we ensured that it was within the interval of 10 minutes, and then we took out the recordings that corresponded to the time block of the point count for analysis. So 15 minutes recording that was simultaneous with the 15 minutes point count was what we used, and then we compared bird abundance and richness across the various sampling techniques. We also estimated the expected species richness from the average of A's, Kayo1, Jackknife 1, and bootstrap estimators using the SPADE R package in R. Then we used the ratio between observed and expected species richness to estimate sampling effectiveness for each habitat type. This method has been used before by Fortunbel et al. In 2020 when they sampled understory birds in different habitat types to compare between point count monitoring and then camera and the use of camera traps. So our results showed reviewed 560 recordings from the entire survey, and within these recordings we had 5 Nightjar species recorded. We had freckled Nightjar, we have the long-tailed Nightjar, we had the plain Nightjar, and then standard winged Nightjar, as well as the black-shouldered Nightjar. And our results showed significant difference between Nightjar abundance when we compared the acoustic recorder and the point count, and the difference in species was 0.35, as well as the significant difference you can see it was less than 0.05. But when we looked at species richness, although acoustic recorders had revealed more species richness as compared to the point count, this differences wasn't statistically significant. And so we went further to compare the effectiveness of each method within the different habitat types of the reserve. So for the gallery forest, we realized 66% effectiveness using the point count as against 79% effectiveness using acoustic recorders. For rocky outcrop, we realized 58% effectiveness as against 83% effectiveness. And then for the savanna, our result revealed 63% effectiveness as compared to 73% effectiveness within this savanna grassland. So Um, Generally, the trend of our results will show that there is because yes, when you look at the result, it will show that there is no perfect sampling method, and any of these methods could work. However, when we compare the advantages and disadvantages of both methods and how they apply to a survey or monitoring protocol in various environments, we could easily choose which of these methods will be most beneficial in sampling bird species or in sampling Nightjar populations. For example, we look at the advantages of point count, where we see that yes, abundance data is easy to deal with as against the use of acoustic recorders, where we try to understand, okay, at what point do we say this is a different individual or this is a new individual. We also look at the disadvantages of point count, and we underline observers safety, access to remote areas, and then the need for more personnel as well as high expertise. But when you compare this with the advantages of acoustic recorders, where acoustic recorders could be good for species rich areas, because after recording it you sit back and you could use you could pay attention and then get each of these species. And while there might be some form of distraction during the point count, with the acoustic recorders you can go back and get each of these species without missing anyone. And again, the acoustic recorders is non-intrusive, so while you are in the field there, you can take record of all species, and you don't have to be, or the birds wouldn't be shy of you, or you shy of the birds, either ways. But again, you see that less personnel is required, because all you need to do is just keep the recorders out there, and then you get the recordings eventually, and you analyze these recordings. Again, observer expertise is easily dealt with, because you could get one person to look through and you're not bothered about, okay, perhaps if it was so person who was out there in the reserve or in the forest, he would have seen this or noted this. Again, for the acoustic recorders, it is easier for us to deal with large area and temporal scale simultaneously. But point count, you would need more personnel to be able to handle this. More personnel will be required to handle this. But the big issue with acoustic recording is the data storage. Where do we store all of this data that has been recorded? And how do we go through this? Because it becomes time consuming. But the good news is technology and AI is now being used to deal with the time consuming aspect of it. But the data storage continues to be a big issue when we use acoustic recorders. So from all of this, we see that the acoustic recorders are a better tool, as they can be used over long periods and across large spatial areas simultaneously, particularly in areas where we have security concerns and there is limited access to remote or study areas, and then we have limited field personnel. However, we understand some expertise that have to come with using acoustic recorders, because again, you have to deal with analyzing these records and doing them effectively. So our results shows all of this, and looking at the implication of this for conservation, we know that we can definitely employ the acoustic recorders. We can definitely employ the acoustic recorders to use specifically for monitoring surveys or monitoring programs across habitat types, across large spatial area. As when we look at the effectiveness, we saw what the result looks like. This study wouldn't have been possible if not for the contributions of Yitma Hope and Danjuma Philibus Dami, Uf Ottosin, and Adam Shaxda, who supervised this work to ensure the quality this work has, and Yitma, who spent time listening to all of these recordings, and then the work was generously funded by Leventis Foundation Nigeria. And thank you so much for listening. I hope that you continue to enjoy the conference. Thank you.
Fantastic. We're grateful to Inunam for sending that presentation and putting that together for us. Inunam is not in the conference presently, so we can perhaps provide some contact information if anyone would like to follow up with him. But we will proceed now to the third presentation within this session. This presentation will be done by Lucy Mitchell, one of our own here on the steering committee. Lucy's affiliated with the University of the Highlands and Islands in the UK, and this talk is titled "In the Middle of the Night: Hidden Markov Models Reveal Nocturnal Movement Patterns and Lunar Phase Interaction in the European Nightjar." And we will get that presentation started for you in just a moment.
Hi, my name is Lucy Mitchell. I'm based in the UK, and I am going to talk about the last chapter of my PhD, which was about Nightjar movement using GPS units. I'm going to talk about hidden Markov models to partition behaviours. So we know that animals forage in places and at times to gain the maximal amount of energy for the minimum amount of expenditure. But some animals are constrained. They're constrained by physical traits, or they're constrained by environmental conditions. And insectivorous species are also constrained by their prey, particularly nightjars. Think about moths. They occur spatially and temporally, quite sporadically, and they fluctuate according to the weather and also the physical environmental conditions. So species are having to find ways to forage more efficiently in order to work with that sporadic nature. So species can compensate. They can forage more when conditions are good, so that they can prepare themselves for times when conditions are quite poor. They are often guided by environmental cues. Nightjars are visual predators. We know that when they have increased visibility, this means better conditions. We also know that nightjars respond to the lunar cycle, and species, including nightjars, have developed strategies to capitalize on profitable conditions. This is called the reduced effort hypothesis. So species will feed more when it's good, when the conditions are favorable, than when they are poor. Unfortunately, we know that humans are modifying the environment, and the modification of the environment can magnify some of the constraints, and they can change the cues that animals are guided by in order to find the more profitable conditions. So it's really important for us to understand what conditions influence foraging behaviour, when and where are the profitable situations. Unfortunately, as with other nocturnal species, we have very little understanding of nightjar foraging. So it makes understanding this kind of change in behaviour with environmental conditions even more difficult. Luckily, we have got really good GPS units now that can enable us to model behaviour at quite fine scales. And this allows us to run some behavioural models called hidden Markov models. These take the GPS data and they model the unobserved behaviours that lie behind the GPS data. So they interpret unobserved behavioural states which is what makes up the GPS track that we can see, the points and the lines between the points. So for example, you might have a short distance between points and they might turn on quite a sharp angle. This might mean that they are resting or they are moving about in a very confined area. So yeah, this gives us very fine scale movement information. So we're asking the question, what causes their immediate behaviour to change out when they're actually moving? What means that they change from resting to foraging, foraging to travelling behaviour? So I wanted to look at this. So I collected GPS data from 41 individual birds over 4 breeding seasons. And I collected data every 3 minutes and also every 5 minutes. Because of differences in foraging behaviour between the sexes that I found in other chapters of my PhD, I modelled the sexes separately, and I also modelled the timing, the GPS timing separately, so a model for 3 minutes and a model for 5 minutes. And once I'd got base models, I added covariates in to describe the environment. So the first thing with hidden Markov models is to parameterize the base model. So we give the model information about the movement behavior of the birds with no covariates. We can plot the raw data. So this is data from one individual bird. So we can see histograms of the step length, so the distance between points, and also of the turning angle. And we can see that this individual has a really high number of very short step lengths. And we can use this to teach the model or suggest to the model the number of behavioral states that we think that there are. Once we have our hidden Markov models and they have information that we're happy with, and we add covariates in, we can then use the states, the behavioral states that have been assigned to the model to populate some other models, in this case generalized additive models, to look at different aspects of foraging behavior. This is what the data look like when the states have been assigned. You can see that the GPS data has been colored according to the state type. This allows you to see visually where the bird is foraging or traveling or resting. So after I ran models for the males, the females, and the 2 different times, the 2 different GPS fixed rates, this is what I ended up with. So firstly, when I ran male and female models, males have more behavioral states than females. They have 2 states that are intermediate type states, which I think differentiates between foraging behaviour but also the territory defence behaviour that males do that females don't do. But you can see that all the birds, males and females, have a resting stage, a foraging stage, and a travelling stage, which for the males is state 4, the far right column. And the resting state's generally under 10 meters between GPS points, and then this sort of intermediate one, which is maybe 20 to 40 meters between points, and so on. What we also know from running these models is that the base model with no covariates is not the best model. Covariates are always better than no covariates. It helps to have covariates to describe the movement behavior, and in these models, lunar variables and NDVI, which is a measure of vegetated-ness, they are the most important variables. Assigned to the hidden Markov model with the lowest AIC, I extracted the states that had been assigned to the model, and I created a variable which was the percentage foraging points per day for each bird for each day that they were tracked. And this was my response variable in a generalized additive model, a GAM. My independent variables were the key covariates from the hidden Markov models, so lunar altitude, lunar illumination, and NPVI. I modeled these as potential predictors of the amount of foraging per day per bird. I modeled them alone, and also I modeled an interaction between lunar altitude and lunar illumination. Because previous work in my other PhD chapters showed very high individual variation in this population, I included individual as a random effect. So the best model actually modeled, it showed that lunar altitude and lunar illumination were additive factors. They were additive variables. I've kept the model with the interaction though, because it was less than 2 AIC higher worth than this best model, and I think the interaction between altitude and illumination is quite important. These are the results from this best model, which shows that individual is indeed significant. The interaction between lunar altitude and lunar illumination is almost significant and quite clouded by individual. I do think, however, it's very important to have individual variation in there, and we can see that it explains nearly 41% of the variation. This shows you the relationship between altitude, lunar altitude, and lunar illumination. This is an interactive heat map. But the terms don't influence the model equally. Remember that the additive model was very close to the interactive model. This is what the two covariates look like when modelled additively. Got altitude on the left, which is slightly quadratic, and then illumination on the right. Because there's so much individual variation, this does cloud overall patterns. So it's quite hard to see general trends, but we know that these are really important variables for nightjar foraging. So to summarize, nightjar foraging behavior is very complex. Males are not very different, but different from females in their behaviors. It's hard to tell maybe display behavior, territory defense behavior from foraging. What causes nightjars to forage more during one night? What are the important variables? It's quite hard to say, but we know that altitude, lunar altitude, and lunar illumination are important here, and we can see from the combination of hidden Markov models and GAMs that foraging is responsive to local fine-scale cues, so behavior can change during the night, but it's also influenced more broadly. The mean foraging, the foraging per night is affected by mean values of the various variables per night. So there's different scales of kind of foraging behavior. And I think it would be really useful to combine not just have the GPS data, but also have accelerometer data to try and pick out particular nightjar behaviors in even more detail. Thank you for your time.
All right, excellent. Thank you very much, Lucy. We have a couple minutes here. Yeah, we have a couple minutes here for questions, and we've got one right off the bat from Mark. Mark says, I enjoyed your talk. Where was your research carried out? Saw the map. Sorry if I missed that fact.
No. Oh, there we go. Right,
There you
can
are.
you hear me?
Yeah.
There we go. I have two different mute buttons on my computer. That's very confusing. So my research was on a national nature reserve in the north of England, so in South Yorkshire. For those of us in the UK, it's quite near to a place called Doncaster, so it's a big flat landscape with lots of peatland habitat in.
Great. We've got one here from Ruben. Did you explore possible influences of habitat differences between or within sites on accuracy and performance of the HMMs?
Yeah, it's a good question. So I explored the influence of habitat on the behavior change. So as well as using NDVI, I actually categorized habitat based on some UAV, some drone photography, and I looked at the influence on the sort of the changes, but I didn't do too much kind of validation of accuracy really, and I think it's quite it is quite complicated because NDVI doesn't always match up properly with kind of habitat categories. You can get all sorts of values of NDVI within one habitat category. So there are quite a lot of complexities there that I think are probably worth exploring a bit more. I expected habitat to be a more influential variable than it ended up being actually. So I think there's more sort of testing to do.
Great. And you had a question from Mark Brigham, but he said that your answer to Ruben satisfied his question. So
fine, fair enough. That's fine.
And we have time for just one more quick question here. So if you would enhance the HMMs with, I'm losing it, jumping away from me with some of the answers. With the accelerometer data, which behaviors would you most hope to distinguish from each other? There we are.
Yeah, that's a good question as well. So I think one of the things that I found really interesting about running these models was the difference between the males and the females. And so males were the 4-state model with these two kind of intermediate states that should be something like foraging. The 4-state model was so much better than the 3-state model, and there were these two quite distinct behaviors in between the slow resting behavior and the commuting behavior. And I would really like to explore that, because from my field work I see such big differences between the behavior of the males and the females. You know, the males do these big territory defense kind of circuits around, and I think it would just be interesting to see if we use the accelerometer data, whether we could actually say if that's what's happening, because I think hidden Markov models can sometimes decide that there are extra states when there aren't. It seemed very sure about these states, but I think it would be good to kind of confirm that difference there.
Yeah, great. Okay, so I do see there are more questions coming in, so feel free to respond to some of those great questions that we're getting in the chat. We're going to move into our 4th presentation of the morning. Thank you very much, Lucy. Our 4th presentation, this next presentation is done by Juan Esteban Salazar from the University of Chile and the Chilean Bird and Wildlife Observers Network. The title of the talk is Invisible Touch, Visual and Tactile Complementation of Nocturnal Prey Capture in the Band-Winged Nightjar. And we will start that for you in just a moment.
Greetings, everyone. It is a great pleasure for me to be here presenting part of my PhD work in this Nightjar community. My name is Juan Esteban Salazar, and I'm from the University of Chile. And I'm going to be talking to you about the work that we did describing the visual and tactile systems of the bandwidth Nightjar. In this talk that I've called Invisible Touch. I'm going to let you know why in this talk. So first of all, as many of you know, nocturnal birds face many challenges related to the low light availability in their environments, and they overcome this by several strategies. First of all, many species have an increased sensibility of the visual system, larger eyes and structures like that, adaptations like that. Other species forego the prevalence of the visual system and rely mainly on other sensory systems such as touch and olfaction, as the case of this kiwi. And many other species complement one sensory modality, the visual system, with another kind of cues, sensory cues, such as the hearing or the smell or the tactile aspects of their behavior. Nightjars are quite special, as we know, and have been kind of neglected in this regard because we know little about the visual behavior and their underlying neural structures that are related to this. As you might have seen in the field, when the nightjars are hunting their prey, they attack from below, and this has been postulated to be related to the visual anatomy of this group, as you can see in the image below, because their large eyes are dorsally oriented, and pursuit starts when a flying insect crosses this visual path. But in the final stages of behavior, of foraging behavior, you can see that the eyes become partially obstructed because this wide mouth opens and the eyes are facing backwards, are not facing directly into the prey. So this suggests that maybe there's another sensory modality that has to be involved in this behavior in order to direct these final stages of prey capture. So we are interested in trying to understand what happens in this moment of capture behavior. We propose that visual and tactile structures complement each other in a dorsoventral way. That's what we call the dorsoventral complementation hypothesis, in which we try to assess this question by proposing that the dorsal area of space is covered by the visual system, and it is responsible for prey tracking, and it's complemented by the tactile system that might be based on the rectal bristle system that is pointing ventrally. So it is ventrally oriented. So this is a part of a kind of wider questions that we have been addressing. We are interested in knowing which sensory modalities are relevant for foraging in nightjars and what are the neural mechanisms that underlie this behavior. And we are trying to understand this working with the bandwidth nightjar, the gallina ciega común, that it is a widespread neotropical species, and also it is present here in Chile. So I'm going to present to you, I'm going to show you some results from this work. First of all, we analyze and describe the visual anatomy of this species, and we were particularly interested in understanding the anatomy of the binocular visual fields that have been shown to be very important in prey tracking and hunting behaviors. So we analyzed the extension and orientation of this binocular visual field, as you can see here in the left corner. And shown here in brown is the extension of this visual field. We found that it is dorsally oriented. And this is quite unusual in birds because most of birds of prey and other species that hunt on the fly have a binocular visual field that is pointing straight, not dorsally. So here you can see the point of the beak in falcon species and in the Nightjar, and you can see that the visual field is dorsally oriented. Also, we analyzed the retinal anatomy, describing the distribution of the retinal ganglion cells, and we found that the maximum density, as you can see here in the top left corner, of these cells is found in the ventrotemporal quadrant of the retina. That is the area that receives visual information from above. Also, we analyzed the anatomy of the optic tectum, that is a midbrain structure that receives projections from the retina and has a spatial map on it, on this of this retina projecting to the tectum, and we found that this dorsal area that corresponds to the ventrotemporal quadrant is enlarged in this optic tectum, also suggesting that there is a visual dorsal specialization. And the other half of this hypothesis is the ventral side. So we wanted to know, first of all, if the necessary structures for mechanoreception were present in the base of each rectal bristle. So the first thing that we did was to describe the histology of this, the anatomy of this tissue. And we did several cuts and stainings of this tissue surrounding each bristle, and we found that there are many mechanoreceptory structures, the Herf's corpuscles, distributed laterally around each bristle follicle. The rectal bristles are marked here in these white arrowheads. And here's a detail of these structures. But also we wanted to know how these structures project to the brain, and in particular to the posterior brain, to the areas that have been shown to be relevant for tactile behavior and related to the face in several species. So we injected some tracers, some neural tracers in the base of these bristles, and analyze where did these projections end in the posterior brain. And we found that many of them end in the trigeminal system. That is one of the most important tactile systems in relation to the face in several vertebrate species. Here on the left, you can see a sagittal section of this brain of the Nightjar. And in particular, in the posterior brain, we see that the areas belonging to the trigeminal system have or receive projections from the rectal bristles. And also we did an experiment where we injected different stainings with different colors in different positions of the bristles. Some were in the rostral end and some in the caudal end with different colors. And here on the right, you can see that when we did this, we found that each bristle projects to a particular place of this trigeminal nuclei, for instance, the principal nucleus of the trigeminal nerve, showing or suggesting that these projections are segregated. So there is at least the basis for a somatotopic map, for a map that we show here, quite similar to what we see, for instance, in the barrel cortex of mice in relation to their vibrissae. So we, as I told you, we injected these different stainings, and you can see here that those bristles project in a partial segregated way in these two pathways, one ascending pathway in the trigeminal system and one descending pathway. And they conserve this partial segregation along the way. So in conclusion, our results are consistent with this multisensory complementation scenario I show you the dorsal ventral complementation hypothesis, in which the dorsal field, the dorsal area of the world of the nightjars is covered mainly by the visual field, by the visual structures, and this part of the space and this sensory system is mainly responsible for triggering the pursuit and prey tracking. But in the end, in the final stages of foraging, these eyes become obstructed, and there's when the tactile system based on the Herf's corpuscles takes in and becomes the main sensory structure involved in this behavior, independently of vision. So we are still wanting to address these open questions. We are interested in understanding how prey is detected and followed, and we also want to know if there are other sensory modalities such as olfaction or audition that can be relevant to foraging in this species. We are also trying to describe the ecology of the bandwidth Nightjar, because it's a species that we know very little about their natural history, particularly here in Chile. And also we would like to try to understand if there's some kind of role of color perception or contrast perception in these scenarios. Finally, I would like to acknowledge all the people that made this work possible, in particular the members of our lab and the field station where we developed this study, our funding sources, and our team, Máximo Fernández, Pedro Fernández Aburto, Macarena Faundes, and Jorge Podosis, that is the head of our lab. Thank you very much.
Yeah, thank you very much, Juan, for that excellent presentation. Just all of the complexities that you're looking into are really fascinating. It was great. We have time for one question here. You've got a question from Mark. So here we are. So would you predict the mouth would only open when the bristles are close enough to the prey to touch it? Meaning, is there no blind spot between vision and touch?
Thank you so much, Mark, for your question. I think that is one possibility among many others. We haven't been able to describe that critical point with the mouth becomes open. We attempted to do some behavioral studies in order to describe that with enough precision, but we weren't able to do that because of the pandemics. But I think that's one of many possibilities. And the other one is that this movement is kind of ballistic. I mean, prey tracking is mainly visually guided, and then when the prey is located close enough, the mouth opens, and there's no need for some kind of visual input because the movement is kind of automatic. But we don't know. We are open to analyze that.
Excellent. Thank you again, Juan. You are continuing to get some more questions in the chat, so feel free to respond to those. Yes, that would be excellent. Okay, so we are moving into our final presentation of this first session. The final presentation is presented by Jitse Cremers, who's a master's student at the University of Antwerp in Belgium. And the talk is titled, "How do various nocturnal light sources drive the perching orientation during foraging activities?" We will play that for you in just a moment.
Good afternoon, everyone. Over the next few minutes, I will share insights from our ongoing research on the impact of light pollution on the foraging behavior of the European nightjar. Nightjars are visual predators, mostly active during twilight periods. They perch at the edge of open fields, utilizing vantage points to spot flying insects against the bright sky. Their perching strategy often involves choosing locations with protective elements on one side, possibly to safeguard against predators, while the other side remains exposed to the open sky. When they detect a prey item, they'll leap into a distinctive parabolic flight to capture it. The reliance of nightjars on a bright sky for prey detection is underscored by previous studies, which revealed their dependence on the lunar cycle. As illustrated in this left graph, their activity aligns with twilight periods, and nocturnal activities are strongly influenced by the availability of light. This activity diagonal mirrors the lunar cycle, with the moon's position affecting their foraging behavior during different moon phases. Notably, nightjars use moonlit skies to forage even during the night. Our research expands beyond lunar cycles. Previous studies linked nightjar activity on moonless nights to cloud cover. Under pristine skies, cloud cover led to darker nights, as evidenced by the green line in the left graph from a study site in Mongolia. However, in our Belgian study sites, where the skies are heavily light polluted, flight activity increased on cloudy nights. Completely overcast, moonless nights can turn as bright as those during a full moon. Nightjars often seem to select perches that shield them from the supposedly moonlit part of the sky, raising the question of whether they orient themselves towards bright sources of light pollution to detect prey. To test this hypothesis, we employ GPS and accelerometer tags to pinpoint foraging sites. The left graph depicts accelerometer data from a foraging individual, and clusters of these events at specific locations were identified as foraging sites. Subsequently, all sky images were captured from these sites, offering a bird's-eye view with protective cover on one side and an open sky on the other. Currently, these images are being analyzed using specialized software to measure sky brightness and identify sources of light pollution. Through analyzing all sky images, we aim to understand how both direct and indirect light pollution might impact nightjar foraging. This left image shows the sky brightness index, and on the right is the correlated color temperature, or CCT. This area here represents sky glow originating from the city of Houthalen, which is a significant source of light pollution at our study site in Meeuwen. This analysis suggests that nightjars may deliberately position themselves to have strong sources of light pollution within their field of view, likely to enhance their ability to detect flying insects. While these initial insights are intriguing, drawing definitive conclusions requires further analysis. So in the coming months, we plan more rigorous examinations to comprehensively understand how light pollution influences nightjar foraging. I look forward to sharing more conclusive findings with you in the near future, and I want to thank you all for listening.
Excellent. Thank you very much for your presentation. I'm trying to confirm whether or not Yitsa is here for questions. Yep, I think okay, I see you now. Okay, apologies. Yeah, so we have time for a couple of questions. Okay, so we've got a question from Ruben here. Ruben says, do you count for moonlight presence when taking your pictures?
Yeah, so we have two different conditions where we take the picture in a condition where the moon is present, so in full moon, for example, and then one condition where the moon isn't at new moon. And then we hope to compare these two conditions and see how it influences their foraging behavior.
Great. Okay. If there are any other questions, we can put those into the chat at this point. And thanks again, Yitsa, for your presentation. And that, let's see, actually, here we go. We got one more time here. So we hope to where are we? Okay, it's jumping away from me. There we go, Lucy. Okay. So do you think you could measure the prey availability and amount in these areas to see if this differs and matches with their behavior? i.e., are there more moths where there's more sky glow? Great question.
I have not included this in the study that is ongoing now, but there are some studies that are also doing this on moths, on their prey species. So I certainly hope to link this research with other ongoing research and see possible trends in that area. Yeah.
Yeah, great. It seems like there's always more variables with this work. There's always something else to consider. Okay, well, thanks again for your presentation. And with that, that concludes our first session of the day. And we are going to enter into a break period until okay, so we are going to resume after our break here. I am excited to get going with this plenary talk, but before we begin, just because we have a handful of new participants that have joined throughout the morning, I just want to touch on just a few things. If you open the chat, we are posting some of our steering committee members are posting some important links relating to the meeting for providing feedback and for contributing to our small grant program. I also want to let you know that you can enable closed captioning if that's helpful to you by using the if you click on more at the bottom right of your screen and selecting to turn on captioning. And we've been recording this presentation and we'll make it available within a week or so. So if you missed any of the talks this morning, you'll have the option of viewing those later. Also, if you would like to suggest some topics for our discussion sessions at the end of the day, you can put those into the chat. All right, so with that, we are going to move to our plenary talk presented by Carlos Camacho. Carlos is a Nightjar researcher with decades of experience and who started, I guess, in reviewing his presentation, inadvertently started a long-term monitoring project focused on nightjars. He's currently tenure track at the Dunya Biological Station in Seville, Spain. And this talk is titled "Doing Long-Term Research on Nightjars and Not Die in the Attempt." So something that I think a lot of us can benefit from listening to today. So we will start that for you in just one moment.
Good morning, afternoon, evening, everyone. Thanks to the organizers for the opportunity to give this plenary talk. I'm so happy to see so many faces from all over the world. To strengthen the global Nightjar network commitment to equity, diversity, and inclusion, I proposed the steering committee to give a plenary talk in a language different from English, and they were all happy to accept my suggestion.
Seems like we might be having a technical issue here. Hold on just one moment.
Good morning, afternoon, evening, everyone. Thanks to the organizers for the opportunity to give this plenary talk. I'm so happy to see so many faces from all over the world. To strengthen the global Nightjar network commitment to equity, diversity, and inclusion, I proposed the steering committee to give a plenary talk in a language different from English, and they were all happy to accept my suggestions, so I'm going to shift to Spanish. After all, a large proportion of Nightjar species live in Spanish-speaking countries across South America, so this might not be the last time you will need Spanish to approach Nightjars. All the slides and subtitles are in English, so please don't panic. En la charla de hoy voy a hablar sobre las oportunidades y los desafíos que representa realizar estudios a largo plazo en los chotacabras, y para ello voy a estructurar la charla en dos partes. Primero me voy a presentar brevemente y explicaré lo que son los estudios a largo plazo, dando una definición y una descripción general de su valor y las dificultades que conllevan. Y en la segunda parte voy a centrarme en los chotacabras y discutir cuáles son las principales dificultades que yo mismo he encontrado haciendo investigación a largo plazo en estos organismos, desde problemas de financiación hasta cuestiones de bienestar personal. Voy a empezar presentándome y explicando las razones por las que elegí el estudio a largo plazo con chotacabras como el tema de esta charla. Soy de España, en la Unión Europea, y me licencié en biología justo después de la crisis económica global de finales de los 2000. España contrajo una enorme deuda que sumió al país en una crisis económica, política y social muy grave, y la financiación para la investigación y las becas predoctorales desaparecieron durante unos años. Así que después de un par de años decidí montar mi propio estudio y financiarlo acerca de la ecología espacial de los chotacabras. Esto lo hice en el Espacio Natural de Doñana, que es un santuario para las aves migratorias localizado en el sur de España. Conseguí una pequeña beca de investigación de la Sociedad Española de Ornitología, compré una docena de radioemisores y pedí ayuda a algunos compañeros y amigos para desarrollar un estudio que planifiqué para que durara solamente 2 años, pero nada más lejos de la realidad. Poco después de que empecé este estudio, conseguí una beca para hacer una tesis doctoral, pero en un tema muy diferente a la ecología de los chotacabras, así que este proyecto pues quedó como un estudio paralelo durante los próximos 10 años. 15 años después, aquel estudio a corto plazo todavía está activo y ha llegado mucho más lejos de lo que esperaba inicialmente, gracias en parte o en gran medida a la generosa colaboración de toda la gente, de amigos y familiares, a supervisores y estudiantes que han hecho que este proyecto sea el suyo propio. Muchas gracias a todos. Debería empezar definiendo lo que es un estudio a largo plazo y lo que no es, y una definición posible para un estudio a largo plazo podría ser un procedimiento que se desarrolla durante un periodo lo suficientemente largo para documentar un fenómeno que tiene lugar a una escala temporal larga. Claramente, la parte más subjetiva y cuestionable de esta definición es cuánto es lo suficientemente largo. Los estudios a largo plazo son muy variables en longitud y su duración mínima pues, es muy difícil de determinar. Posteriormente volveré a este problema durante la segunda parte de la charla. De momento vamos a centrarnos en el valor de los estudios a largo plazo, o dicho de otro modo, ¿por qué necesitamos estudios a largo plazo? Muchos cambios ambientales y muchos procesos ecológicos y evolutivos tienen lugar durante periodos prolongados de tiempo, que van desde años a décadas o siglos. Realizar estudios a largo plazo es esencial para documentar estos cambios y para entender los procesos biológicos que requieren largos periodos de tiempo. Algunos ejemplos de estos procesos son el aprendizaje, la transferencia cultural, la senescencia o la microevolución. Los estudios a largo plazo son también fundamentales para replicar los experimentos y protocolos y así verificar la generalidad de los resultados y patrones que se observan en estudios a corto plazo. Basar estudios en medidas repetidas de los mismos individuos es también fundamental para separar los papeles que tienen los procesos que ocurren a nivel poblacional de aquellos que ocurren a nivel individual. A partir de los estudios a largo plazo es relativamente fácil inferir tendencias y patrones que podrían no detectarse si el rasgo o los procesos de interés fluctúan marcadamente a lo largo del tiempo. Por último, pero no menos importante, un beneficio importante de los estudios a largo plazo es que permiten a los investigadores establecer una conexión entre la causa y el efecto en procesos que tienen lugar a distinta escala temporal. Ejemplos de estos procesos son, por ejemplo, la preferencia de hábitat inducida, como el imprinting, la expresión de caracteres fenotípicos dependientes del contexto, efectos epigenéticos y otros muchos. Pero si los estudios a largo plazo son tan informativos y tan positivos, entonces, ¿por qué son tan raros? Bueno, pues porque no todos son buenas noticias. Los investigadores que coordinan estudios a largo plazo tienen que hacer frente a un importante número de desafíos y obstáculos que pueden llegar incluso a un fallo en el estudio. De todos estos desafíos, quizá el primero y más evidente se relaciona con una financiación discontinua. La razón para esto es que los financiadores han priorizado generalmente metas a largo plazo, en lugar de los beneficios que conllevan la investigación observacional a largo plazo, creando así una tensión entre lo que significa estudiar un sistema no perturbado a largo plazo frente a desarrollar experimentos a corto plazo. Los problemas de financiación nos llevan al punto 4 de la lista, ya que los estudios observacionales a largo plazo no son siempre compatibles con manipulaciones experimentales que alteran el curso natural de las poblaciones. Pueden existir también interferencias entre preguntas de investigación que serán más frecuentes a medida que nuevas personas y nuevos objetivos se incorporen a los estudios a largo plazo. Para aliviar los efectos no deseados de estas interferencias, lo que yo recomiendo es definir al principio del estudio cuáles son los protocolos basales o los datos que representan la columna vertebral de nuestro estudio. Los estudios a largo plazo son difíciles de implementar para la gente joven debido a la inestabilidad laboral durante la etapa postdoctoral. Sin embargo, algunos investigadores senior podrían ser ya demasiado viejos para empezar un estudio a largo plazo y desarrollarlo durante un tiempo suficientemente largo como para ganar inercia. Espero que nadie se sienta ofendido. Yo, para ser sincero, admito que ya me siento demasiado viejo para mantener funcionando nuestro propio estudio. Bueno, pasemos al punto 3. Ahora mismo, los científicos se encuentran bajo una presión sin precedentes para publicar y así poder tener éxito en su carrera académica. Si nos adherimos al principio de publica o muere, entonces mejor que no empecemos un estudio a largo plazo porque recolectar datos suficientes para un solo paper lleva tiempo, a veces incluso años. Además, los estudios a largo plazo requieren de un diseño cuidadoso que permita mantener los protocolos y las metodologías estandarizados y constantes en el tiempo, al menos todo lo posible. Ya que cualquier cambio en los protocolos podría comprometer seriamente la validez de comparaciones con periodos anteriores de tiempo. Para evitar este problema, yo recomiendo revisar y adaptar, si es necesario, los protocolos de campo durante los primeros años y, por supuesto, pedir ayuda a investigadores con experiencia. Cambiar de observadores no es bueno, pero mantener a los mismos observadores tampoco está exento de problemas. Cuanto más sepamos sobre nuestra especie o nuestra población, mejor será nuestra capacidad de recoger datos. Por ejemplo, las tasas de recaptura podrían aumentar durante un estudio a largo plazo simplemente porque somos mejores atrapando a nuestros animales después de que practiquemos durante varios años. Para minimizar los efectos de esta fuente de sesgo, yo recomiendo hacer algunas pruebas piloto antes de empezar un programa más ambicioso y a más largo plazo. Un piloto rápido nos permitiría anticiparnos a problemas futuros, especialmente si planeamos trabajar con especies o poblaciones muy poco conocidas, como es el caso de la mayoría de los chotacabras. Cómo se manejan los datos es también un importante desafío en los estudios a largo plazo. ¿Cuándo o dónde vamos a almacenar nuestros datos? cuál debe ser la estructura y el formato de nuestra base de datos, quién debe ser responsable de la digitalización, cómo combinamos la información de distintos años variables o protocolos, necesitamos registrar datos adicionales como la identidad del observador. Pues estas son algunas de los ejemplos del tipo de preguntas que surgen antes de empezar un estudio a largo plazo. Y es importante porque años de esfuerzo y de perseverancia en recoger los datos pueden quedar anulados si no se toman los datos de forma y se almacenan de forma eficiente. Este hecho se sumariza, se resume perfectamente por la frase de un colega que dice que hay dos tipos de personas, aquellos que ya han perdido datos y aquellos que los van a perder. Así que, por favor, ser muy cuidadosos con el almacenaje de datos. Bueno, pasemos a los chotacabras. A lo largo de las próximas diapositivas voy a tratar de explicar qué es un estudio de chotacabras a largo plazo y discutir desde mi experiencia personal una selección de los desafíos más importantes que los investigadores que trabajan con chotacabras se encuentran cada día o cada noche. Mucho de lo que sabemos realmente sobre la ecología y evolución de las aves está basado en un pequeño grupo de especies que han atraído la atención de los investigadores, principalmente porque son fáciles de capturar y seguir, como aquellas que crían en cajas nido. Sin embargo, un número tan bajo de organismos puede generar preocupación sobre cómo de representativo son estos organismos de la ecología general de las aves. Así que es importante considerar también organismos no modelo. La mayoría de los miembros de la familia Caprimuridae se conocen muy poco y son buenos candidatos para investigación a largo plazo porque son relativamente longevos. El problema es que los estudios más largos hasta la fecha duran apenas una década. ¿Esto es realmente largo, lo suficientemente largo para que se pueda considerar un estudio como a largo plazo? Pues retomando la pregunta que dejé sin responder al principio de la charla sobre la mínima duración de estudios a largo plazo, hay básicamente dos criterios principales. Uno, el tiempo de generación o la esperanza de vida de la especie modelo. El segundo sería utilizar la duración media de otros estudios en el mismo campo como guía. Atendiendo al tiempo a la esperanza de vida, al criterio de esperanza de vida, un problema potencial es que los chotacabras son longevos y, por tanto, es importante seguirlos a lo largo de mucho tiempo para entender de forma completa sus historias vitales. Pero mucho peor que esto, la mala noticia es que para la mayoría de las especies ni siquiera sabemos cuánto viven. Existen datos sobre esperanza de vida para un puñado de especies y parece que viven al menos tanto como se han estudiado. Lo que no sabemos es si estas estimas serían más largas si continuamos recogiendo datos o si usamos métodos de captura-recaptura para estimar su esperanza de vida. Es posible que sean más largas, pero realmente no lo sabemos. Así que para determinar la duración mínima de un estudio a largo plazo en chotacabras, yo me decantaría por el criterio más pragmático de la duración de otros estudios. Los estudios a largo plazo en especies longevas, como las aves marinas, pueden ser realmente largos, tanto como la vida de un hombre, tal como demuestra esta bonita secuencia de fotografías del profesor Dunnett y el Fulmar 57, uno de los individuos que siguió a lo largo de su vida durante más de 40 años en las Islas Orcadas. Por suerte o no, no existen estudios de chotacabras que hayan durado tanto tiempo, así que la duración mínima de los estudios en este campo en particular, es relativamente corta. De hecho, la mayoría de lo que llamamos estudios a largo plazo no deberían ni siquiera llamarse así, ya que son simplemente una serie de estudios independientes que comprenden diferentes protocolos dependiendo del estudio. Por lo que yo sé, los estudios más largos hasta la fecha en caprimúrgidos son los del chotacabras europea en Reino Unido y Bélgica, liderados por Greg Conway y sus colaboradores y Ruben Evans, respectivamente, y nuestro propio estudio sobre chotacabras cuellirrojo en España. Aún así, la duración total del estudio más largo es solo de 15 años. La mayoría de los estudios son más cortos o se han desarrollado por menos de una década. Así que Considerando que la investigación en chotacabra es una disciplina relativamente joven, cualquier estudio que se desarrolle durante más de 5 años podría considerarse como un estudio a largo plazo. Parece algo bastante factible, ¿no? Sigamos con otro desafío. Asumamos que un estudio de 5 años, pues nos lo podemos permitir, pero es muy probable que la historia natural de nuestra especie modelo pues no se conozca. Los investigadores han evitado estudiar a los chotacabras principalmente por su comportamiento críptico y su actividad nocturna, y por tanto no es sorprendente que aspectos fundamentales de su biología todavía no se conozcan y sean un misterio, limitando la base para los estudios a largo plazo. Para empeorar las cosas, mucha de la información disponible se ha recogido en Europa y Norteamérica, por lo que no sabemos o podría no ser representativo del amplísimo número de especies que crían en regiones tropicales. Así que antes de abordar preguntas grandes, y dependiendo de la especie de estudio y la región, podríamos necesitar dedicar los primeros años de nuestro estudio para recabar información sobre aspectos básicos de la ecología y el comportamiento de nuestra especie de estudio. Yo personalmente me tomé este periodo como una oportunidad para recoger datos de historia natural, así que lo disfruté mucho esta fase. Por ejemplo, uno de los primeros papers que salieron de nuestro estudio a largo plazo proporcionó las primeras estimas para cualquier especie de chotacabras de parámetros tan básicos como la edad de primera reproducción, la tasa de reclutamiento o la distancia de dispersión natal. Este ejemplo ilustra bien los desafíos únicos a los que tienen que enfrentarse, en particular los investigadores en chotacabras. Otro desafío es el cambio del paisaje. Muchas especies de chotacabras crían en ambientes transformados que están sujetos a continuos cambios. En la mayoría de los casos, los investigadores no podemos hacer mucho para evitar esto. Así que no queda otra opción que utilizar estos cambios en el paisaje como una oportunidad para poder aprender sobre las respuestas de los chotacabras a las presiones antropogénicas. Por ejemplo, nuestros chotacabras cuellirrojos utilizan las carreteras como plataformas de alimentación y se sientan en la superficie enfrente de arbustos relativamente altos y densos que están en el borde de la carretera y que los protegen de ataques de depredadores. Aquí tenéis una fotografía de uno de nuestros chotacabras sentado sobre la superficie de la carretera, justo al lado de un olivo que está en el borde. La eliminación de la vegetación de cuneta en algunos tramos en nuestra área de estudio ha resultado en un cambio en la distribución local de los chotacabras que se alimentan, y una reducción del número de chotacabras que utilizan las carreteras que están cerca de los territorios de cría, ya que necesitan buscar sitios alternativos para poder alimentarse. Pero quizás el principal desafío de realizar investigación a largo plazo en aves crepusculares o nocturnas, como los chotacabras, sea la falta de sueño. De todos estos problemas, este es quizás el más difícil de resolver, porque no queda otra opción para los investigadores que salir ahí fuera durante la noche a capturar o a seguir a los chotacabras. Dormir bien es importante para evitar un montón de problemas de salud. Y se asume por lo general que dormir 7 u 8 horas es lo mínimo requerido para tener un funcionamiento óptimo. ¿Pero realmente esto es posible? Pues yo realmente creo que no, al menos no en el día a día. Yo me di cuenta muy pronto y no me quedó más remedio que asumir que no podía funcionar de una forma óptima durante la mayor parte de la estación reproductiva de nuestro campo, sino de una forma más bien basal. El problema es que más a menudo de lo que me hubiera gustado, simplemente no pude dormir ni siquiera para funcionar de forma basal. Y como podéis imaginar, el problema aumentó a medida que mi fitness inclusivo también aumentó. Estos ojos rojos e irritados después de una larga noche de captura de chotacabras y de llevar a mis hijos al colegio, nos llevan al siguiente desafío: encontrar un buen balance entre la vida y el trabajo. La vida es difícil de por sí, pero esta dificultad puede ser incluso mayor para investigadores que hacen el turno de noche. De todos los desafíos que se han considerado aquí, el impacto del trabajo de campo nocturno en la familia es uno que, en mi opinión, requiere una atención especial. ¿Hay algo que podemos hacer para reconducir nuestro balance de vida y mejorarlo? Pues por supuesto, pedir ayuda. Si algo he aprendido yo durante los 15 años que he estado desarrollando investigación en chotacabra de forma continuada, es que no puede desarrollarse un estudio de este tipo en solitario. Durante mi carrera he trabajado con paseriformes, saltamontes, salmones, topillos, alcaravanes e incluso pinzones de Darwin, entre otros, pero el modelo de estudio que realmente ha causado un impacto mayor en mi vida normal son los chotacabras con diferencia. Quizá porque son los que más me importan o quizá simplemente por el hecho de tener que ir al campo durante la noche. Pero hay algo que está claro. Nuestro estudio a largo plazo hubiera sido mucho, mucho más corto si no hubiera tenido la ayuda en estos años de tanta gente brillante que me ha acompañado. Otro reto es la financiación. La financiación es la bestia negra de los estudios a largo plazo en general y los chotacabras no son una excepción. La mayoría de las entidades financiadoras pues, priorizan metas a corto plazo, como expliqué al principio, y además hay una preferencia quizás inconsciente por animales graciosos o grupos de animales que nos resultan familiares o nos inspiran curiosidad o ternura. Desde luego los chotacabras no están entre estos y la situación puede ser incluso más difícil para hablantes en español, ya que uno de los términos, uno de los nombres en español de los chotacabras, se parece mucho al de una criatura mitológica muy famosa de México, el chupacabras, y como resultado mucha gente se confunde y ni siquiera cree que nuestro organismo de estudio exista. Yo no puedo pensar en un escenario más frustrante y peor para conseguir fondos que este mismo. Algunos de los estudios o muchos de los estudios a corto plazo que finalmente se convierten en un estudio a largo plazo nacen durante estadios tempranos de nuestra carrera y durante esta etapa las oportunidades para conseguir financiación son limitadas a pequeñas cantidades de dinero de entre 1 y 3.000 dólares. Entre 1.000 y 3.000 dólares, por lo que es necesario acudir o intentar conseguir un apoyo adicional, especialmente si se van a utilizar tecnología de seguimiento o análisis moleculares. El merchandising de productos relacionados con el proyecto nos dio una estupenda oportunidad para conseguir fondos para hacer el trabajo de campo. Nuestra primera experiencia fue esta bonita camiseta que difería de las clásicas camisetas de pajareros en que su diseño no era, en que la diseñadora no estaba realmente interesada en los pájaros y quizás por esto pues hizo un dibujo abstracto de un chotacabras, a diferencia de las imágenes típicamente realistas que hacen las delicias de la mayoría de los pajareros. Y en segundo lugar, otro aspecto poco usual es que ofrecíamos tallas y formas diferentes para chicos y chicas. Yo creo que la buena calidad artística del diseño y esta forma sexualmente dimórfica de nuestras camisetas dispararon las ventas de las primeras 300 camisetas simplemente con hacer un post en nuestras redes sociales. Este potencial, pues obviamente no debía desperdiciarse, así que pronto creamos un logo basándonos en el nombre original del grupo de investigación, Nightjar, y lo usamos para la segunda camiseta, que una vez más pues se vendió la primera tirada muy rápidamente hasta el punto de que tuvimos que hacer tiradas cada vez más grandes. La segunda y la tercera se vendió muy bien y en conjunto ahora mismo pues puede que haya más de 1.000 camisetas distribuidas por todo el mundo literalmente, tal como demuestra este collage con los múltiples sitios en los que nuestros colaboradores se han tomado fotos. Para celebrar la primera década de nuestro proyecto, organizamos además un festival de un día de duración, al que vinieron 200 personas que pasaron allí el día comiendo, bebiendo y bailando. Pero esto no es la única opción para financiar nuestra investigación. Más recientemente, incluso he escrito un pequeño cuento para niños, que a diferencia de las camisetas o el festival, pues no pretendía ser una fuente de financiación, sino una herramienta simplemente de divulgación. Aún así, para cubrir los costes de producción del libro, lo anunciamos en nuestras redes sociales y como resultado vendimos más de 150 copias solamente en las 2 primeras semanas. De forma intencionada, el libro pues cubrirá los gastos de la temporada de campo de 2024. Así que, bueno, pensad y ser creativos. Todo esto está bien, pero los beneficios son generalmente insuficientes para cubrir los gastos de desafíos de investigación más ambiciosos, ¿no? Que conlleven la colocación de aparatos digitales, análisis moleculares. Si no estáis todavía en la posición de pedir fondos por vosotros mismos, quiero decir, de ayudas grandes y cuantiosas, pedid colaboración. Los investigadores senior quizá no te den el dinero, por supuesto, pero si encontráis un interés común, ellos pueden daros pues, aparatos, cubrir los costes de análisis moleculares u ofrecer un contrato para un técnico de campo, por ejemplo. Yo he recibido realmente ayuda muy valiosa del centro CAMMOV, el Lund, especialmente en la forma de data loggers para seguir a los chotacabras durante el ciclo anual. Otros investigadores, como el profesor Mark Brigham, también ha sido muy amable conmigo y me hacía revisiones muy suaves, pero muy sesudas, cuando yo publicaba mis primeros papers como único autor, a pesar de que tenía muchísimos defectos. Quiero daros las gracias a todos, Mark, Susan, and Anders, if you are there listening to me, thanks a lot. Y para terminar, pues bueno, quería poner otra vez énfasis en el enorme potencial que tiene el hecho de colectar o recoger datos estandarizados durante periodos largos de tiempo y el valor añadido que conlleva hacerlo usando especies no modelos como los chotacabras. No niego en absoluto que hacer investigación a largo plazo en los chotacabras es largo y que mantener un proyecto de investigación a largo plazo es incluso más desafiante. Sin embargo, a pesar de todos estos contratiempos y las limitaciones de financiación, el proyecto actual produce un paper SCI cada año en promedio y aproximadamente unos 20 estudiantes han completado ya sus proyectos usando los datos que generamos en nuestro proyecto. Así que el balance de momento ha sido muy positivo. Os deseo la mejor de las suertes en vuestros propios proyectos. Larga vida a los chotacabras y muchas gracias a todos por escuchar. I'll be happy to answer your questions in English if you want.
Thank you very much for that presentation, Carlos. There were so many useful insights there that I think would benefit, all of us in this group, wherever we stand in our careers and our studies with Nightjars. We are unfortunately out of time for live questions here, but Carlos is here and available to respond to questions. So I hope that you'll take him up on that in the chat. There's so much that could be discussed based on that presentation. So thanks again.
Thank you. Thank you.
Okay, so with that, we're going to move into the second half of our day, the conference, and I am going to hand over my hosting responsibilities to another individual on our steering committee. So I will see you guys for tomorrow's talks. Thanks again, and I'm going to pass things on now to Richard to cover the second half of the day. Thanks very much.
Yeah, thanks for introducing me, Logan. So welcome to the second half of the first day of our conference. And we will proceed with the second session on natural history. And the first talk will be covered by Paula Hidalgo Rodriguez. She's a postdoc researcher at the University Pablo de Olavide and Estación Biológica de Doñana. So from the same research group as Carlos, actually. And the talk title of her presentation is Survival Rates of Juveniles and Adults of a Nocturnal Migratory Bird. And we will play that for you just in a moment.
Hi everyone, this is Paula Hidalgo, and I'm going to talk to you about the survival rates of juveniles and adults of red-necked nightjars. Studies on demography are quite important in order to know the processes and structure of natural populations and the mechanisms underlying this process. Most studies on avian demography are made with adults, but only few are made with juveniles, and almost none with fledglings and birds in earlier stages. So the great importance and influence that could have these stages on later development of the individual. In addition, there is a global decline in insect abundance, which is having serious consequences for insectivorous bird populations. Therefore, to investigate the demographic processes that occur in early stages of insectivorous birds can help to establish efficient conservation measures. To fulfill this knowledge gap, we have assessed the survival rates of adults and juveniles red-necked nightjars, many of them captured as fledgling individuals for the first time. Red-necked nightjars are nocturnal migratory birds that breed in the Iberian Peninsula and North Africa and spend the winter in Southwest Africa. They are seed and weight foragers that feed on aerial insects, mainly moths. In the last decades, they have suffered a decline of 30% in their Spanish population. For this reason, we have calculated the interannual survival rates of juveniles and adult red-necked nightjars. The data used to run the analysis comes from a long-term monitoring study of a breeding population in Doñana National Park in southern Spain that started 15 years ago. We calculated the interannual survival rates of nightjars for the period 2010-2021. And as a result, we got that juveniles have lower survival rates, around 35–40% than adults, around 66%. But still, they were very high, with no temporal variation among years. The differences between sexes are almost negligible. For recapture rates, it seems that it's easier to recapture an adult of age 1 that was born last year in the same breeding area, with an almost 50% of probability to recapture, than to recapture older individuals, which recapture probability is of 35%. Also, we check for the possible influence of environmental covariates on survival probability, but none of them, precipitation, NDVI, or temperature, seem to have an effect. So, for conclusions, nightjars have high survival rates, even for juveniles, which makes them resemble the life story of seabirds. Also, it looks like nightjars are quite resilient at changes in weather and climate through these last 10 years. Although, it would be interesting to evaluate these last 2 years, 2022 and 2023, because they have been extremely dry in southern Spain, but when the analyses were made, this data was not ready. In addition, we think that juveniles have quite high survival rates compared to other species because of the long period of parental care. From our radio tracking monitoring data of these 2 last year fieldwork, we found support for this hypothesis because we saw how nightjar chicks could stay within the same parents' area until they are almost 50 days of age. Thank you so much for your attention.
Great, thanks Paula for your talk, and I've seen that she's here with us in the meeting, so maybe we have a time for one short question. Yeah, so Lucy asks, thanks for the interesting presentation, and she agrees that works on juveniles is so important. So what do you think? Why are there no obvious temporal variations in survival rates of juveniles and adults?
Thank you, Lucy, for your question. Well, maybe they are as much resilient that they do not get the influence of temporal of environmental covariates on their survival rates. But maybe, as I say in the talk, if we could have take into account these last 2 years that we got a very extremely dry situation in South Spain, maybe these survival rates change. But yeah, I mean, it also like shocked to me. But I have to say that in a previous study, also done in the same population, but like 15 years ago, they got the same survival rates. They did not have a temporal variation in our red-necked nightjars.
Yeah, fascinating. Thanks for the answer. And there is another one from Maria, but please answer it through the chat because we'll have to move forward with the next talk by Brianna Zafiro. Oh, sorry. Thank you very much, Paula. And then we can move to the second talk by Brianna Zafiro. She's a master's student at Nova Southeastern University in the USA, with the talk title, The Gastrointestinal Endoparasites of the Cryptic Nightjar Family in Southeastern Florida. So let's hear the talk.
Hello, everyone. My name is Brianna Zafiro. I'm here today with the Fisheries and Avian Ecology Lab and the Parasite Ecology Lab at Nova Southeastern University. I'm talking about the gastrointestinal endoparasites of the nightjar family in southeastern Florida. Parasites can reveal trophic interactions that might be missed by traditional food web studies that are based solely on diet. Knowing parasite and host systems can provide key information on host ecology and food web dynamics. Many parasites have indirect life cycles involving 2 or more hosts. These indirect life cycles can show relationships of hosts being predated on by different hosts. Parasite data, however, is lacking in many birds, especially the cryptic ones without bright feathers. There is currently very little known about endoparasites in nightjars. Most are from chance individuals. There are only 5 species with documented endoparasites. These species are from Brazil, European Russia, the Philippines, and South Florida. One of these records are from the Iberian Peninsula, documenting a capomulga form with a parasite from the order Cestoda. So these descriptions may not even be at the family or even the genus level. Nightjar knowledge is remarkably low compared to knowledge on other groups of birds. This information is from select bird groups in Florida, from the Forrester and Spalding's Parasite and Diseases of Wild Birds in Florida. Most groups have more surveyed species, if not the same number of species of nightjar endoparasites that are known globally. Birds were collected frozen from partnering wildlife rehabilitation centers in South Florida. Specimens were thawed and dissected for endoparasites. The GI tract was examined, including the esophagus, trachea, proventriculus, ventriculus, intestines, and cloaca, as well as the washes of the proventriculus, ventriculus, and intestines. These helminths were extracted, stained, and mounted. Samples were then identified by standard taxonomic keys in the Parasite Ecology Lab's library. The study species of these surveys are the Chuckwell's widow, the common nighthawk, the eastern whippoorwill, and the lesser nighthawk. The Chuckwell's widow has 55 individuals. The common nighthawk has 19 individuals. The eastern whippoorwill has 7 individuals. The lesser nighthawk is a vagrant along with the Antillean nighthawk. However, we have only gotten 1 lesser nighthawk, and since we do only have 1 specimen, the lesser nighthawk will not be included in the analyses later in this presentation. The prevalence of endoparasites do vary by species. The Chuckwell's widow has a 47% prevalence rate. The common nighthawk has a 58% prevalence rate, and the eastern whippoorwill has a 29% prevalence rate. This shade plot shows intensity by species across host. The Orthoscrigibinia and Raeliantina species do have the highest intensity. The Subularis sectoria is seen in all 4 species, including the lesser nighthawk. Almost every endoparasite was a host record. Each name with a star next to it is a host record. Along with these host records, a range expansion was seen in Subularis sectoria, a nematode previously described in Cuba. The host records of this project and the Chuckwell's widow are the Chaliobella species, the Subularis sectoria, Liposomum species, Orthoscrigibinia species. In the common nighthawk, it is the Stomilotrema vicarium species, the Raeliantina species, the Subularis sectoria, and the Orthoscrigibinia species. In the eastern whippoorwill, the new host records are the Chaliobella species, Subularis sectoria, Stomilotrema vicarium, and the Subularis species. In the lesser nighthawk, we only did get a Subularis sectoria, and this is a new host record. So these results do add to global knowledge with two more species with described parasites, 14 new host records, as well as 1 range expansion. In total, the results total out to 7 nightjar species, as well as 22 endoparasites described from the nightjar family. Along with these results, parulids have been found in the proventriculus of the Chuckwell's widows. Parulids, commonly known as warblers, were found in 6 Chuckwell's widows, 5 during the project in the Fisheries and Avian Ecology Lab, and 1 by Yaritza Acosta at the Pelican Harbor Seabird Station. The most recent warbler is suspected to be an American redstar due to the orange feathers in the proventriculus content. This behavior has previously been reported by Thayer in 1899, describing a Chuckwell's widow catching a warbler midair while on a ship, and Ower in 1966 reporting a deceased Chuckwell's widow and with a warbler in the proventriculus as well as the mouth. And so to conclude, the infection rate across all southeastern Florida nightjars is 47%. This is intermediate with other bird families in the area, as insects are not common intermediate host. However, ground nesters may have a chance of more parasites. Based on this study, we propose endoparasite community composition reflects both trophic behavior and nesting strategy. Further examination is needed as nightjars are a vastly understudied family for endoparasites. Lastly, we're thankful for the continued support from our research partners at South Florida Wildlife Center, Pelican Harbor Seabird Station, Key West Wildlife Center, and the Florida Keys Wild Bird Center. We also wish to thank Kathy Afena-Tapiche from the Miami-Dade College and Kaylee Craddock for their preliminary assistance, and the Homos College of Arts and Sciences for its continued support. Thank you.
Thanks, Brianna, for a really insightful presentation in this very important and often really undercovered topic. I think that Brianna should be here, so we have time for 2 or 3 questions, so feel free to post them in the chat. Yeah. Alicia asks that you have recorded presence of parasites, but did you happen to notice much variation in parasite load in your specimens?
Hi, so we did look at the variation of parasite load, and so that's what the intensity of parasite infection is. But the bird that had the most or the highest intensity was the Chuckwell's widow. We believe that because this bird is a little bigger and has a little longer of a GI system, that's kind of gives the parasites more space to attach in like the intestines and the proventriculus and whatnot.
Yeah, sure. Thanks for answering the question. And in the meantime, we have to wait for another one if there's any. So feel free to ask. We have 2 more minutes for the discussion for this talk. Okay, so if you can think of any more questions, please post them in the chat, and I hope that Brianna will address them. And thus we can move to the final talk of this session on natural history before a break. And this session by Haruka Mizumura, a researcher from Mount Fuji Research Institute in Yamanashi Prefectural Government in Japan, considers a great nightjar and how it breeds in semi-natural grassland on the foot of Mount Fuji, Japan.
Hi everyone, I introduce nightjars in grassland in Japan. This is female and here's a chick. Thank you to everyone who helped my work. The great nightjar, Yotaka in Japanese, their breeding range decreased 57% between 1970s and 2000s. Almost all of the nightjars studies focused on forests and logging areas. There are only 2 breeding records in grasslands. Therefore, the ecology of nightjar in grassland is unknown. In addition, grassland area is only 1% in Japan. However, some grasslands are remaining in Mount Fuji like this. So, grassland and endangered species conservations are critical issues. We conducted nightjar survey in this burned grassland in several years. Today we focused on 2 questions: how many nightjars breed there, what environmental features relate this species density. This is survey site. We placed 10 fixed points with 250-meter radius in this burned grassland. This grassland has 2 geological features. The first is volcanic fern deposit, gray area in this figure, and the second is a rubble area, white area in the figure. Volcanic fern deposit is plain area like this picture, and rubble area is ridged area like this. We conducted playback survey in each point and playing nightjar score like kyo in twice in each point 2 years. The number of maximum males was recorded as the number of territories. These are results. Territories are located mainly in rubble area like this fixed points, and there was positive correlation between the number of territories and rubble area. These are brief conclusions. How many nightjars breed there? There are many nightjars in the burned grassland. What environmental features relate this species density? I think rubble area seems to be important, but further consideration needed. Rubble area and volcanic fern deposit are a very different color like this. There are some questions to consider in the future. We will continue this research. Thank you for listening.
All right, thanks for the presentation, Haruka. It must have been very intense, especially the morning surveys from 1:30 a.m. So I got the information that Haruka is here and ready to do a live Q&A session. So please feel free to post your questions into the chat. We have 3 minutes, approximately 3 minutes for discussion. So Becky asks, did you identify nest sites?
Yes, but only 5 nests, but all nests located in rubble area.
Okay, thank you. And there's a second question from Lucy. Is the Lowell area warmer? Does the ground absorb more heat than might explain the territories or more nests located in that area?
I don't know the rubble area warmer than explained earlier, but I think yes, it is future consideration, and
yeah, of course, it might be really interesting to explore why they are preferring these areas with lava.
yeah, I think so. Yeah.
Ash asks, without regular burning, do you think this lava habitat will be in danger? Or is there any other mechanism that regulates the extension of such habitat?
Yeah. Yeah, regular burning is important, I think, because the without burning, the grassland habitat maybe change or forest area.
Oh, yeah, I see. Good. Are there any more questions on Haruka? Yeah, probably not. So thank you very much for giving the talk and also answering the questions. So this concludes the session for the first session for natural history, and now we'll insert a 10 minutes break, and we will meet at 13:35 UTC. So see you in 10 minutes. All right, welcome back, everybody. We shall continue with the final talk session as of today of the Global Nightjar Network online meeting. The session 3 is considering, again, natural history, but also taxonomy. But before we start with the talks, please, especially the newcomers that have joined us, that has joined the meeting now, please see the chat where you can find all the information regarding code of conduct and also regarding feedback on our meeting, and also links where you can buy some merchandise as a Redbubble t-shirt, and also you can donate for a small grants program run by Global Nightjar Network. So let's proceed to the first talk of session 3 by Jose Manuel Zamora Marin, who is affiliated with Miguel Hernandez University of Elche in Spain, with a talk uncovering life history traits in the family Capremulgidae, breeding dispersal, and mate fidelity. So we shall hear the talk in a minute.
Hello, everyone. First of all, I would like to thank the organizing committee of this interesting initiative. I'm Jose Manuel Zamora. I'm currently working as a postdoctoral researcher at the University of Miguel Hernandez of Elche in the southeastern of Spain. I'm going to talk about some life history traits poorly studied in the family Capremulgidae. Particularly, my talk will be focused on assessing breeding dispersal distance and the mating system in the red-necked nightjar. Well, as most of you probably know, aerial insectivorous birds are declining worldwide, and this is mostly as a result of some anthropogenic threats, such as agricultural intensification and habitat degradation. And this is a scenario forces us to implement effective conservation actions focused on mitigate these threats. However, conservation programs also require a deep knowledge on the species life history traits to maximize effectiveness and resource allocation. For example, if we know which stage is constraining reproductive performance across the entire annual cycle, we can implement specific management actions within that stage to improve the breeding success and ultimately to enhance long-term population viability. One of these reproductive stages is the mate selection, which comprise all questions related with the mating system. This reproductive stage, the mating selection, may have direct implications on breeding performance and even adult survival. For example, previous studies conducted with bred geese have highlighted that faithful geese, faithful individuals, showed a higher survival rate than those individuals which divorce and establish new pair bonds. In this context, this result could be explained by the fact that partner rotation, being faithful, may reduce sexual conflicts and energetic cost devoted to the courtship. Thus allowing to allocate more time, for example, to foraging, and ultimately leading to an increased survival rate. Up to today, we know that the vast majority of bird species around the world, about 90%, show long-term social monogamy, for which a breeding adult retains the same partner as long as this partner is alive. But despite this overall pattern, there is also a small suite of bird species which show short-term social monogamy. For example, plovers and other shorebird species. However, the vast majority of studies on avian breeding biology have been focused on a restricted suite of model species. So the current theories in ecology and evolution are based on this suite of model species that in general correspond to colony breeders, nest box breeders, and threatening species, because these species are easy to monitor in the field, and their nests are easy to be found. But what's about the remaining portion of avian diversity? By focusing only on model species, we are overlooking a lot of life history traits, which are sometimes exclusive to single avian orders. For example, these are some of the most poorly studied avian groups in the world, for which data on breeding biology are extremely scarce. And as you can see, nightjars comprise one of these groups. Well, just as a brief introduction, the family Caprimulgidae is comprised by about 100 species distributed across the world. But despite the widespread distribution, breeding biology of nightjars remains poorly studied for most species. For example, information on clutch size, on incubation period, or the chick rearing period is lacking, is absent for about 75% of the known nightjar species. And even for those nightjar species with available data, studies usually rely on a too small sample size of nests. But why there is scarce information on nightjar breeding biology? The reason is because it's very, very difficult to find the nest in the field, not only in this type of habitat, on densely vegetated habitat, for example, in the UK, but also even in open habitat. This is a picture of our study area. This is the ground under the citrus trees. And as you can see, roosting and incubating nightjars can camouflage very, very good in this type of ground. Well, our project focuses on the red-necked nightjar, Caprimulgus guficolis, which is a long-distance migrant that breeds in the Iberian Peninsula, but also in northern Morocco, and moves in autumn to the wintering grounds placed in the south of the Sahara Desert. Our study area is placed in the province of Murcia in southeastern Spain, which is a semi-arid region, a Mediterranean semi-arid region, where irrigation agriculture has recently extended to almost half of the surface area. Despite the widespread decline of the red-necked nightjar in other Spanish regions, the species seems to be nicely adapted to this highly transformed agricultural landscape, which may even support a great density of breeding nightjars. And this is a detailed picture of our study area, which has about 5 square kilometers, and is mostly comprised by citrus plantations, which are used by nightjars for nesting, but also by sparse Mediterranean shrublands and vegetable crops, which are used by nightjars for foraging. Here we are conducting a regular breeding monitoring since 2017 up to today. The main objectives of this study were to determine interannual breeding dispersal distance, to estimate between year mate fidelity and divorce rate, and to assess sex and fidelity related difference in breeding dispersal distance. For that purpose, we conducted nocturnal visits aimed to look for nests with the help of a hand light, and well-located nightjar nests were regularly monitored at daytime to know the hatching date. And when chicks were between 5 and 15 days old, we used a mobile mist net to trap both parental adults at each nest and market them with official metal rings. To this way, we could gather data on individuals' life history for each captured adult, and we assessed whether each nightjar retained the mate between 2 consecutive years or divorce and breed with a new one. In this context, we could find 4 different situations depending on the nature of the available information. For example, we could speak properly about interannual mate fidelity when both breeding adults have been captured in the same nest within a given year, and they are found breeding again together in the next year. On the other hand, interannual divorce occurs when an adult breeds with a new partner different from that the previous season, despite the previous one is still alive and is also captured in the current year. Well, these 2 scenarios, mate fidelity and divorce, can be only identified when both partners of a given nest are captured in 2 consecutive years. However, this is not always the case, because sometimes we are unable to trap both adults at each nest during consecutive years. For such case, then we can't distinguish between true divorce and widowhood, but is the death of a partner. Well, moving to our preliminary results from 2017 to 2020, we found 191 nests of red-necked nightjar distributed across our study area, and we could capture both breeding adults in 110 out of the total sample size. when we assessed the breeding dispersal distance of the captured nightjars, we observed that most of them returned for nesting within 300 meters buffer from the nesting site of the previous year. There were a few birds that moved longer distance, up to 600 meters, and 2 single individuals move about 1 kilometer away. Therefore, these results support the high site fidelity of nightjars already provided by previous studies. And when we assess difference between male and females in the breeding dispersal distance, we actually observe that there are no evident difference, because both male and female show a similar pattern of breeding dispersal distance. And this is also the case for the fidelity degree. When we assess the dispersal distance between divorced and faithful birds, we don't find evident significant difference in the pattern. Lastly, we explored the mating system of the red-necked nightjar by estimating the rate of fidelity and divorce between consecutive years, and we found that most nightjars were faithful and made with the same partner year after year. However, we also recorded some particular case of divorce for which we had evidence of an adult mating with a different partner despite the previous one was still alive. We couldn't know the identity of a given partner for a portion of the monitoring breeding pairs, about 35%, because at least one adult was not captured due to logistical limitations. Then, in such case, we can know whether they correspond to true divorce or new partner mating following the death of the previous partner. In summary, we can conclude that social monogamy is the main mating system in the red-necked nightjars, because about 88% of the captured adults retained the partner from 1 year to the next. Overall, the breeding dispersal distance was too short, and nightjars rarely move longer than 300 meters for nesting between consecutive years. And what is now the core question of our research is to determine the direction of the influence. That is, which factor is the cause and which factor is the consequence. There are some policy studies highlighting that mate fidelity acts as a determinant factor of the site fidelity, but there are also research supporting that the opposite pattern also occurs in some species. In our case, we think that benefits of breeding dispersion are precluded by the high habitat homogeneity, because citrus plantations are quite similar across the entire study area. Then mate fidelity could be acting as a determinant factor of site fidelity in our nightjar population. Of course, all this work is not only mine, it's the result of a long-term collaboration established between wildlife biologists and ecologists from the Southeastern Naturalist Association in Murcia and the Nightjar research team in Doñana. I would like to thank all the volunteers that have helped during all this time. And that's all I wanted to share with you. Thank you so much for your attention, and I'm available for any question. Thank you so much.
All right. Thank you for another very complex, but indeed interesting research on red-necked nightjars. We have a couple of questions here, so we can proceed to answering them if Jose is here to answer them live. The first question is, I have observed strong site fidelity in common nighthawks, but only if they were successful nesting. If the nest failed, they did not return to the site. Did you observe that?
Hi, thank you so much for the question, Becky. Really, we didn't include the data of the breeding success in the analysis because the results are still so preliminary. But just like a personal observation, when they fail, when they fail the breeding attempt, they used to try again very close to the previous attempt. But we want to include to add in the models the breeding success of the previous attempt to test this question.
Sure, thanks for the answer. And a second, or actually two questions in one, by Iraida. Do you know if breeding dispersal was influenced by previous breeding failure or / success? And just a curiosity, do you know if there is extra pair paternity? Sorry if you said it and I missed it.
Okay, thank you for the question, Iraida. Just more or less the same. We are now exploring the data to assess if there is any influence or from the breeding success of the breeding attempt of the previous breeding attempt. And regarding to the extra pair paternity, we took samples in collaboration with George Day some years ago to test to assess if there is extra pair paternity in our population. But at the moment I don't know the preliminary results. It's unpublished still.
Right, yes, so we'll be looking forward to final results when you'll publish it. There is another question, but we'll have to move to the next presentation. So thank you again very much, and please address the question in the chat. So the second talk of the session 3 is by Thiago Vernacci Vieira da Costa, one of the members of our steering committee. He's a professor at University Federal de Itaúba in Brazil, and his talk is titled Revision of Family Group Taxa and a Proposal of Adjustment to the Higher Level Classification of Caprimulgidae. So the stage is yours.
Hello, everyone. My name is Thiago Vernacci. I'm from Brazil. I'm from the Federal University of Itajubá. And I'm gonna present you here a revision of family group taxa and a proposal of adjustment to the higher level classification of Caprimulgidae. The family Caprimulgidae traditionally used to include two subfamilies, which were Cordeilini and Caprimulgini, and this classification used to be based on the presence or the absence of rectal bristles around the beak, crepuscular versus pure nocturnal habits, wing shaped, if they are more pointed or more rounded wing, and mainly, especially based on the characters of the palate. So Cordeilini used to be mentioned to present a desmognathous palate, which you can see here, where these two processes, they are closed above the vomer bone, while in the Caprimulgini, the palate used to be was mentioned to be schizognathous, which the vomer do not contact the two maxillopalatine processes. So this is used to be the traditional classification, and the genera that used to be included in these two subfamilies used to be these here. You can see. So these two authors they converge and include some genera, but they do not agree in some others. So clear 1998 included Lurocalis, Cordalis, Nyctiprogny, and Podager in Cordelini, and all the other genera in Caprimogini, and Holyoke in 2001 included Eurostopodus, Cordalis, Lurocalis, Nyctiprogny, and Velis, and Cordelini, and all the other in Caprimogini. So this classification remained until very recently. However, recent molecular studies converged and showing that these two subfamilies, they are not monophyletic. So here there's a compilation made by White et al. 2016 showing different authors as Klug et al., Larsen et al., Hand et al., Sigurdsson et al., and White et al. 2016, showing that the two subfamilies, they are not monophyletic. Actually, Cordelini is indeed monophyletic, as you can see here, expressed as nighthawk. So there's a clade nighthawk in all these phylogenies. Nighthawk, this clade is Cordelini, but all the other clades are included in the traditional Caprimulgini. So based on this phylogenies, so we need to make a higher level revision of this classification. So in this phylogeny here, what we have is what we see if we put all those phylogenies together in agreement. So we come out with this phylogeny, so showing that Cordelini, the nighthawks, they form a monophyletic group, and Caprimogeny includes all the other lineages, these other groups. So we need to make a revision in order to reestablish monophyly. Therefore, after these studies, I consulted and I visited many different collections to study the anatomy of the different groups of Caprimulgi in order to try to find some important characters in the skeleton of the species. So I visited 14 institutions in Brazil and in other countries, and I studied 9060 skins, 255 skeleton specimens of 49 species, comprising all those groups that I presented before. And based on those phylogenies and based on the morphological variation that I noticed among those lineages, among those groups, I present here a new proposal of classification of the Caprimulgini. So instead of having two subfamilies, I suggest three subfamilies, which are Eurostopodini, as you can see here, Lincornitini, which would be a new subfamily in the family here, and this blue box here, as you can see, would be the subfamily Caprimulgini. So the traditional Cordelini are now included in this subfamily Caprimulgini. So this is a new proposal of classification regarding subfamilies. However, I found also important variation between those clades included in what I'm calling here Caprimulgini. So in order to recognize this variation, I suggest also some taxa in this subfamily Caprimogini, which I proposed as tribes. Therefore, what you can see here is the phylogeny of Caprimogini based on all those molecular phylogenies. And here's some proposals of tribes inside this subfamily Caprimogini. So we have 5 clades, 5 lineages in this subfamily. So I propose here 5 tribes corresponding to these clades, to these lineages. Therefore, we have a clade of Old World species, which the name Caprimogini is available and would apply. We have here the clade of the Nighthawks, which the name Cordelini is available and would apply. We have here the clade of the Purwell and Alice, for which there is no available name. So one suggestion would be Antrostomini. It would be a new tribe. There is a big clade here of South American species, for which the name Nocturnal is available and would apply for this group. And here there's a clade include just one species, one genus, and one species, that is Gactornis ineratus, for which there's no available name in the family group taxa. So here there's a suggestion of a new tribe named Gactornitini. Therefore, I propose here a new classification for the family based on the phylogenies. So in order to establish the monopoly in the group, and in order to recognize the morphological variation, so I propose 3 subfamilies, and in the subfamily Caprimogini, 5 tribes would be included. So thank you very much. I'm happy to be here, and I would be happy to hear your opinions on this new proposal. Thank you so much.
All right, Thiago, thank you for this groundbreaking, indeed, presentation on the phylogeny of and the clades of Caprimulgids. Yeah, it's indeed very interesting for all of the people here to see how it correlates with the current knowledge. So are there any questions on Thiago? Okay, so if you can't think of any questions now, you can always reach out Thiago through the chat or write him an email. I believe that he'll be happy to answer you afterwards. So that means that we can move to the final talk of session 3 by Tom Shannon, who has recently completed his PhD and is currently affiliated with the University of Aberdeen in the United Kingdom, with a very interesting talk referring to an Nightjar, and with a question, what is an Nightjar?
Hi, I'm Tom Shannon from the University of Aberdeen, and my talk is entitled What is the Nightjar Nightjar? Resolving the Taxonomy of Caprimulgus solala. The Nightjar nightjar is perhaps the most enigmatic nightjar species. It is known from a single specimen, a decomposing corpse found on a dirt road in Ethiopia's Nightjar National Park in 1990. When the corpse was disturbed, a large number of feathers were lost to the wind, but a single wing was recovered in good condition. It was also noted that there were white tips to the outer tail feathers, though these too were lost. But with this salvage wing acting as the type specimen, Caprimulgus solala was discovered, with solala translating as only a wing. But what made this wing so distinctive that it could be confidently declared a new species? Firstly, its large size set it apart from other African nightjar taxa, with a wing length of 198 millimeters, with the majority of species falling in the 130 to 160 millimeter range. And secondly was the unusual positioning of the white patch on the four outer primaries, which was significantly higher up the wing than the emarginations on those primaries. It is unlike any known taxa, and yet there have been no further sightings despite numerous dedicated expeditions to the national park over the last 30 years. This brings into question its taxonomic validity. Is it an incredibly rare species in dire need of conservation efforts, or is there something else going on? We decided to investigate this genetically. There are 25 recognized species of nightjar in Africa. We retrieved samples from 52 individuals across 17 of these species, including the Nightjar type specimen. And with the addition of sequences available on GenBank, we were able to account for all species with the exception of a tomboy nightjar, Africa's other single specimen species. DNA was isolated separately from tissue samples from museum specimens, and then from feathers, blood, and buccal swabs from live ring birds to minimize the risk of contamination. The Nightjar nightjar type specimen was then genetically sexed, and we targeted 4 genes, 1 mitochondrial and 3 nuclear, in an attempt to place Nightjar nightjar phylogenetically. Mitochondrial data was retrieved for all sampled species, while 3 species failed to yield nuclear sequence. These 3 species are the brown nightjar, swamp nightjar, and freckled nightjar. Firstly, the genetic sexing PCRs showed the Nightjar holotype produced a single band when visualized on an electrophoresis gel. This shows that it has ZZ sex chromosomes. It's a male. In females, which are ZW, we would expect to see a second band on the gel. We then produced a phylogenetic tree using just the mitochondrial data. Unexpectedly, the Nightjar type falls confidently within the standard wing nightjar clade. It would appear that, at least through its maternal line, the Nightjar specimen is actually a standard wing nightjar, and not its own species. But is this just an aberrant plumage variant, or is there more to the story? Nuclear DNA contains 2 copies of each gene, 1 from each parent. Although diversity in these 3 genes studied was low, a blast search of our dataset showed that only 1 haplotype was consistently a best similarity match for standard wing nightjar, suggesting that only 1 of its parents was a standard wing nightjar. This tells us that the Nightjar nightjar must be a hybrid. However, the lack of consistency between genes for the second haplotype suggests that the paternal species has yet to be sequenced. In all likelihood, it is one of the 4 species for which we failed to recover nuclear data. So who is the father? Of course, cryptic diversity may mean that there are species splits unaccounted for, and these cryptic taxa are missing from our dataset. However, based on the data we have to hand, only 1 of the 4 known taxa missing from the nuclear dataset represents a plausible option as Nightjar's father, as I'll now attempt to show through geographical likelihood, morphometrics, and plumage data. So of the 25 species listed here that are found in Africa, which of those are known to occur in Ethiopia? Of those, only 14 are known to occur in Ethiopia. From these 14, which of the species have white patches on their outer tail feathers? We've removed 4 more. There are only 10 potential candidates left. Of these remaining 10, which species have white patches on the outer 4 primaries? No fewer, no more. Only 8. And crucially, which species have a large wing length? That which is within 10% of the nightjar type. Only 1 of these 8. The remaining 7 are much smaller. And this gives us only 1 candidate that's plausible as the father, the freckled nightjar, and it is missing from our nuclear data set. We can further strengthen the case for freckled nightjar being the father of Nichiza by looking at the relative position of the emargination on the P9 primary feathers, one of a suite of ID features used for separating nightjars in the region. A ratio is calculated as the length from the emargination to the feather tip as a proportion of the total feather length. If we take the mean ratio for standard wing nightjar and freckled nightjar and scale them for the P9 length of the Nichiza type specimen, we can see that the emargination is located much closer to the feather tip in standard wing nightjar, while for freckled nightjar, the emargination would be much closer to the feather base than that of Nichiza. However, if we calculate the intermediate position between the two, as would be expected from a hybrid, it is virtually identical to that displayed by the Nichiza type specimen, differing by less than 1 millimeter. While this body of evidence strongly supports freckled nightjar being the father, only genetics will reveal the truth. And so the mystery continues. So to sum up, what is Nichiza nightjar? Firstly, Nichiza nightjar is not a valid taxon. Genetic evidence shows that it is a hybrid between a female standard wing nightjar and another, currently unsequenced, nightjar species. Based on geographical range, morphometrics, and plumage data, the only plausible paternal species is freckled nightjar. However, to conclusively put this mystery to bed genetically, we are in desperate need of a fresh freckled nightjar sample. I would also welcome any nightjar samples for genetic analysis. If you can help, please reach out. Thanks.
All right, so as mentioned in the chat already, this was indeed a real detective work to find out and conclude this almost 3 decades old mystery, which I think most of us were thinking about the validity of this bird and whether it can be observed and whether it's still out there in Ethiopia. So yeah, it seems that the question itself might be over, but there are still opportunities for you, the participants of our meeting, to ask your questions and to post them to Tom via chat, as he's not open for the live Q&A session. But yeah, please feel free to ask anything regarding the topic of Nichiza or Nightjar in the chat. So Alicia asks, what is do you know if there are any other records of hybridization in nightjars? So we shall wait for the answer from Tom. Yeah, there are a couple of other remarks, and yeah, congratulations on the Tom's work, because yeah, indeed it's really groundbreaking stuff that we are pleased to hear on our conference. And yeah, really looking forward to publishing the final results in the near future. Yeah, so we still have 2.5 minutes before the poster session and the discussion session starts. So yeah, please let me introduce you to Padlet side, where all the presented posters are uploaded by one of the members of our steering committee, Lucy Mitchell. So feel free to reach out and see the posters there. I believe that you will get the link in a second in the chat. And then, yeah, we will proceed with the poster session, which will be succeeded by a discussion session where you can discuss all the information about Niger breeding, ecology, migration, and all the breakout rooms that will be created. Yeah, please note that we are missing 3 posters, but nevertheless, we hope that you will be happy to look at the posters. And there is 1 poster that shall be presented tomorrow at the end of the second day of the conference. In the meantime, there's a second question on Tom. What are the factors that can lead to hybridization in nocturnal bird in their habitat? Indeed, a very important question. Regarding the ecological and maybe also biological factors. Yeah, so Tom has answered the first question that actually there are no other records of Niger hybridization, and he's pretty sure that this might be the first record in Old World Nightjars. So yeah, this is really something that even highlights the importance of this work, and yeah, we'll be very excited to see the final outcome. All right, so the time is up for session 3, Natural History and Taxonomy. So now you shall be should be redirected to the poster and discussion session breakout rooms where my other colleagues from the steering committee, Maria and Tiago, will take care of you and facilitate the discussions in the breakout rooms and give you all the information that you will need for these sessions. So thank you and see you again at wrap up in half an hour. Yeah, so please don't leave, but wait for the discussion for the poster session and discussion breakout rooms to start.
Hello, folks. So we are now in this session, this poster session. As you noticed, we posted the link of the Padlet in the chat. As Richard stated, we have a few posters that the people were not able to upload and to present here, but this time now is time for the discussion. So the people that sent the posters that are available there. They are here. We'll be glad to discuss. So if anyone has questions and comments, please also check in the chat the link for the Padlet to see the posters, and feel free to ask questions for the people if they are here. The authors are here, and everybody are welcome to discuss and to comment and make questions on the posters.
Yeah, just a quick information, we should start the poster session with a video by an artist, Mark Gibbs, and then proceed to other posters in this session. So Gabriel, please play the video for us, and then we'll proceed to the posters itself.
My name is Mark Gibbs. I'm an artist. I'm fascinated by Nightjar. I'm particularly interested in their camouflage. How come they're so good, and their folklore. I've got a piece here. This is an Egyptian Nightjar, and I use birds and other animals to explore history. This game looks at camouflage, and it's a way of introducing an environmental game. It's a way perhaps of slowing people down so that you can have a conversation with them. The winner of the game is the best camouflage bird, and using the window as a light box is a great way of producing the template. It's quite a nice art activity in itself, and you can do this with any subject, and it's a way of starting, and then you can get the participants to cut it out, or you have them already cut out as I've done. So you need some players, and I'd like to introduce you to Helen Walsh and Phil Dutton. The game is to camouflage your nightjar. So you have your blank nightjar here, and we start off with some leaves. They migrate, and this is their habitat in Africa, these leaves and the edges of forests. So first of all, I'm going to ask you, for round one is to camouflage your bird against this background. And what we'll do it for about 4 or 5 minutes, and then your bird is going to fly to a new environment, which I'm not going to tell you what it is. I brought artist oil pastels, and I recommend them. You could do it with coloured pencils or even felt tips. But what I've found is for round 2, you might want to add a white, a light colour, and that adding a light colour over a dark colour in for example coloured pencils isn't so easy. And if you haven't used them before, they are like wax crayons but a higher quality. And this is interesting because there is a study by Stevens et al. That shows that nightjar arrange themselves to precisely fit the background, which shows that you must have self-awareness of their particular patterns. So the winner of round 1 is Phil's. Is it always going to be against this background? It's not. So coming up as a mystery background, you've got the option to stick or modify, but you don't know what you're modifying against. So you have to use your imagination. Right, let's see what mystery landscape we have. We have a forest and a clear winner for round 2 as well. Should we do one more round? Best out of 3?
Why not?
So you can stick or modify. One thing that this game does need is everybody to start together so that the rounds work, so everybody is in sync. The winner is definitely for this is Phil's.
Well played.
we've done 3 rounds. We could go on, but I think you get the idea. But you can see that the one that worked generally has now failed against its specialist habitat. And if we bring out maybe the summer breeding ground, both have something. They're pretty good, but the greens in here are not working so well. So nightjar being ground nesters, they're specialists, and they're much more like this, aren't they?
I think it's a really fun game. It makes you think about how animals have to adapt, and they don't have the option of just popping to a named retailer to change their jacket. They've got to think about the different environments they're going to be in and decide, like Mark was saying, whether they're going to be specialists or generalists. And it's really nice to be hands-on. It's nice to have a game where you're doing something and creating something. I think it's a really good concept.
Yeah, I very much enjoyed it. I was grateful that I got lucky with the habitats. I wouldn't get that lucky again, I don't imagine, but yeah, very enjoyable. And so I have to go home and open a packet of pastels.
Well, that's the Nightjar game for you. I hope you enjoyed watching. I think it's a game of wide adaptability to different animals, and I really enjoyed it. I hope and I think the people who took part did. And isn't it wonderful that Nightjar can bring people together?
Thank you, Gabriel, for the video, and thanks, Mark, for this fantastic work. If someone has any comments and questions, I think that Mark is around here. Mark, if you want to compliment something, if you want to say something, people feel free also to make questions and comments to Mark.
I see there's a question from Mark and I'm wondering the same thing, if there's any chance folks can get that outline to use as well.
Yeah, absolutely. Can you hear me? Okay. Yeah, absolutely. I can put it on. I have an Instagram page or a Facebook page. I can supply that. But the easiest way to do it is to get a photo, maybe from the net, and use that window technique as a light box, and then you can get your own. But certainly I can supply images. I've got a blank that you can use. That's no problem.
Yeah, we have a comment here in the chat that I actually don't know who. Maybe the question is iPhone 7. This comment, really great work, Mark. Please don't really know what Nightjar is. This is a great introduction and activity to what Nightjars face. And this is really interesting, especially for people that don't know exactly what Nightjar is.
Absolutely, I think Nightjar need a PR push. No one I've come across really, apart from ornithologists in the UK, know what a nightjar is. Everybody knows an owl, but nightjars need a bit of PR, I think, because you're not going to conserve an animal if you don't know it. You've got to love it, and you've got to know it to love it first of all.
Mm-hmm. And I have a comment, a question myself. Do you have many children that participate in this exhibit, or they get interested in this art? This is a fantastic educational resource, so I think that it would be great to introduce children in this activity. Yeah.
Absolutely. So I work at a youth centre in inner city Carlisle, and I run it twice with them. So we've run it with about 2 groups of 20 children. Quite a handful to run it with, but it's a good, they all enjoyed it, I think. The main thing I learned from that process is you've got to have a start, got to start them off all at the same time. Otherwise it does get complicated. But I think it has wide adaptability. So you can play it with, I played it with children between the ages of 7 and 12, but you could play it with older children or young people. Or as you see, with played it with trialed it with some artist friends. So I think it has wide adaptability.
Great, awesome. And Maria made even an interesting comment here that for you to consider creating a lesson plan with instructions and materials and post widely, like awayparkcommons.org, for example, for educators to use. That would be fantastic.
Yeah, absolutely. I can do that. What I'll do is there's my contact details. I'm sure we will share them at the end of this, but it's also on the end of that video, and I can certainly put together something like that. I haven't put it together yet, but the great thing is it is quite simple. You just need your template. I recommend oil pastels and laminate your mystery backgrounds. So it's a fairly simple session to run.
Mm-hmm. Okay. Does anyone has any comments? So feel free to put comments in the posters directly in the Padlet, or if you want to ask something to Mark here, you can do here, or you can do directly in the Padlet site. Mark, there is some other questions. So Alicia asked the same about children, so we have already responded. And if someone has questions for Mark, feel free to do here. And please check the Padlet that you can see again the presentation, and you can see also again the other posters. We have 2 other posters for your consulting in the Padlet. Actually, we have 4 posters in the Padlet. You can post questions directly there, and if some of the people is around, they will be happy to respond to these questions.
Okay, thank you, Thiago. I think what we want to do now is open a few breakout rooms to give folks time to chat and talk. Today's breakout session will be shorter, about 15 minutes. Tomorrow it will be a bit longer. We hope to potentially have same topics across the days. And to have the breakouts run smoothly and to be able to share information with the whole Nightjar group here, we ask that breakout rooms have 1 facilitator and 1 note taker. If that note taker could email me the notes for their session, it doesn't have to be anything fancy, just some kind of idea or list of topics that were covered. That would be great, so we can share that more broadly to the group after the conference. There's no experience needed to facilitate one of the breakouts. We just would love to see folks involved. Please introduce yourself briefly to the group, but you have limited time, especially today. Also, if appropriate, we ask folks to discuss some topics like our conservation. What are the conservation priorities for Nightjars related to your topic? What are some other actions or steps related to the species that you all are interested in or would love to see more about? Right now, I've created 4 breakout rooms, one on nocturnal habits, one where it's just a greet and meet, one on breeding, and then another one for poster authors to go ahead and pop in if people want to talk directly to poster authors. I'm going to open those all right now, and you can go ahead and pick which breakout room you would like to join. One of us will stay here in the main session, so if anyone has questions or difficulty navigating, you can pop back and ask us here, and we'll give a little warning before we close the breakout. So go ahead and move to those and have some good conversations. Thank you.
What I wanted to add to the table today.
Yeah, definitely succeeded in that.
So, thank you. Especially, I just, if I'm, I just wanted to thank Carlos, Dr. Carlos, because I'm facing lots of issues being representing an developing country. So there are very limited resources for ornithological research. And when you are talking about nightjar research, it's extremely difficult to find even a 1 penny on the name of research. So today I got lots of answers just because of this presentation, that I realized that, okay, how I can figure out that what can be the secondary source of funding. So
Yes.
thank you for that.
You're welcome.
Well, I just wanted to add one more thing. Hello.
Oh, can you hear me?
Am I audible to you?
Yeah, I
Yeah.
have problems with the connection, I think. Let's try it.
Yeah, Devin, I think that you're frozen for a couple of seconds. So yeah, can you please repeat your question?
Thanks.
Yeah, actually, I was inspired by his talk because lots of funding issues are there in developing countries. So when you are discussing about migration or movement related research, it requires some transmitters or some basic instruments, which is still out of the reach when you are pursuing your career as an early career researcher. You are doing some job to fulfill your research interest, and simultaneously I have a small recently registered NGO in India. So is there any collaborative platform that can provide some instrument for barter system or some kind of collaborative work that can be done in terms of migration or breeding ecology in India?
Well, I don't really know about India, and I don't think there is like an official helper or provider. What I did, I was lucky enough to collaborate with people from Lund University who produce their own devices, and those are the ones that I've been using in my own populations, and we are now collaborating to publish those results. Right, Gabriel? I promise to do that very soon. So I'm not sure about India, but you don't need to find someone there, but you might try to find someone out of India with more resources or shared interest which may fit yours. So he or she could provide some devices to you, and just to answer a question of interest. But yeah, it's hard to find someone willing to provide devices unless they are interested in the same questions as you are.
Thank you for that. And I request all, and I welcome all, whoever is interested in more than 11 species of nightjars. I request all to join hands together. I can accommodate, at least the India can accommodate 11 PhDs together. So please feel free to think about that.
Okay. Keep fighting. Good luck.
Yeah, I guess that we turned this room on nocturnal behavior to more of a meet and greet room, but yeah, it's not not a problem, not a problem, of course. But yeah, if you have any more topics to cover from the nocturnal behavior or nocturnal habits, please feel free to do so, because we have 2 minutes to go until we'll return to the main room. And of course, we can continue discussions tomorrow during the breakout room session as well at the end of the day.
Well, I wonder if we should
Can I?
continue. Oh, sorry if I. Yeah, go ahead.
Hello.
Yeah, we can hear you.
Oops.
Yeah, there seems to be some connection problems again.
Hello. Hello. Am I audible?
Yeah, we can hear you.
Yeah, maybe you can type the
Yeah,
question
okay,
in the chat instead if the problem persists.
Got it. There was a network issue. Sorry for that. The question regarding nocturnal techniques is, I'm totally new to Nightjar research. I came to know this topic came in my understanding from last meet. I was a participant in last Nightjar meet. After that, I realized that there is a huge scope to work on nocturnal behavior of birds. So, are there resources or some techniques or some manuals that can provide information about how a novice or beginner researcher in this field can move forward?
Can you hear me?
Yes.
I think some researchers have their own manuals. For example, I know the one which is called How to Connie, which was developed by Laura Graham, I think. So maybe she's happy to share that with us. I also have our own manual, but it's only describing the measurements we take, but not the regular field protocols. But I'm happy to pass that to you as well.
Yeah, just a quick reminder that the breakout rooms will close in less than 30 seconds. So yeah, please continue with the discussions tomorrow, and yeah, we'll see each other in the main room in 15. Yeah, all right. So that's it for today. We went through beautiful presentations and also poster sessions, discussions in breakout rooms. Yeah, regarding natural history and taxonomy mainly, and also the artistic work of Mark. So yeah, one of the things that I would like to remind is that the meeting today and tomorrow is or will be recorded, and the recording will be shared with you in a few weeks when we process both days of the meeting. In the meantime, if you would like to buy some merchandise of the Global Nightjar Network, such as the T-shirt like this, you can reach out our shop on Redbubble. You can find the link in the chat. And you can also make a donation for the small grants program that we are starting in the Global Nightjar Network. Also, we would be very pleased to get your feedback from how you like the whole conference, and if you have any suggestions for improvement for next year's, or if you want to be a bigger part of the Global Nightjar Network, please let us know and we will try to reach you out as soon as possible. Of course, I would like to thank all the participants and the presenters especially for preparing this beautiful Wednesday sessions that we were able to attend. And we are looking forward to see you tomorrow on the second day of the Global Nightjar Network virtual conference meeting 2023. And we will continue on Thursday, so tomorrow, with the start at 1600 UTC, with sessions covering breeding ecology, breeding and conservation, migration, Nightjar stories, and the conference will be closed with general discussion ending at 2000 UTC. So that's it then. Thank you very much once again, and see you all tomorrow. Goodbye.